Am I audible to all everyone? Hello.
Hello.
A very good morning to all the speakers and delegates present over here. Myself, Jessica Jones, moderator for this virtual session. On behalf of Scholars Conferences, it gives me a pleasure to cordially invite you to the Scholars World Congress on Drug Delivery, Material Science, and Biopolymers. I thank you all for finding time in your busy schedule to be here today. I hope this virtual session will be a remarkable event which will bring researchers, professors, and students in making the virtual session a perfect platform to share experiences and foster relationships. Before starting the meeting, I would like to give some tips for the meeting attendees. First thing, please mute your microphone to keep the background noise minimal. Please mute your microphones while the speaker is presenting. Also, be mindful of your background noise, and please position the camera properly so that while presenting, we cannot see your faces. Also, limit distractions. If in case the mobiles are there, keep it in silent. And also, I request you to avoid multitasking. With that being said, now I would like to call upon our first keynote speaker.
Okay, can I start?
Yeah, let me introduce the doctor. Yeah, sure.
Okay.
I would like to call upon Dr. Peter Chu from National University of Malaysia.
Okay. Can I start?
Yeah.
Okay. Hello, everyone. Today I will be using a video presentation. Hope you all enjoy it.
First, I would like to thank Drug Delivery for inviting me to discuss Peter Chu formula for calculating COVID-19 vaccine efficiency. This discussion, I emphasize the significance of educating the public about the importance of vaccination. By doing so, we can tackle the skepticism surrounding vaccines and improve vaccine acceptance. Article COVID-19 Vaccination Educational App highlights a major issue surrounding vaccine acceptance. People question the safety and efficacy of COVID-19 vaccines. Unfortunately, this skepticism has resulted in low vaccination rates, particularly among certain populations. It is crucial to acknowledge and address these concerns to ensure widespread acceptance and uptake of COVID-19 vaccines. By doing so, we can collectively work towards achieving higher vaccination rates and effectively combat the spread of the virus. Throughout this session, I will discuss strategies to promote better understanding and trust in the efficacy of COVID-19 vaccines. Together, we can pave the way for increased vaccine acceptance. Let's take a closer look at the findings of a survey conducted in Penang, Malaysia, which shed light on the reasons behind the refusal of the COVID-19 vaccine. The survey revealed that a significant number of individuals expressed concerns about the safety of the vaccine, accounting for 75.5% of the respondents. Additionally, 47.4% of the participants cited concerns about the effectiveness of the vaccine as a reason for their refusal. These findings highlight the need for effective communication and education to address these specific concerns. By providing accurate information and addressing misconceptions about the safety and effectiveness of COVID-19 vaccines, we can help alleviate these apprehensions and encourage higher acceptance rates. According to article World Health Organization, Field Evaluation of Vaccine Efficacy, Walter A. Orenstein et al., the ideal vaccine efficacy study is a clinical trial starting with persons susceptible to disease. In a double-blind randomized placebo-controlled fashion, asterisk half of the children receive vaccine and half receive placebo. To calculate vaccine efficacy, both groups are followed prospectively to determine attack rates for disease in vaccinees and non-vaccinees. This type of study is generally not possible after a vaccine has been licensed because the vaccine is of proven benefit, and use of a placebo is unethical. In most countries today, measles vaccine has been used in a proportion of the population. These vaccinees are a self-selected rather than a randomly selected group, and their susceptibility prior to vaccination is generally unknown. Nonetheless, vaccine efficacy studies are still possible if biases are reduced to a minimum in order to recreate as closely as possible the ideal conditions of the prospective clinical trial. General principles vaccine efficacy is measured by calculating the attack rates of disease among vaccinated and unvaccinated persons and determining the percent reduction in the incidence rate of disease among vaccinated persons relative to unvaccinated persons. The basic formula is written as vaccine efficacy equals attack rate in unvaccinated group minus attack rate in vaccinated group, and then all divide attack rate in unvaccinated group and times 100%. Let's calculate the measles vaccine efficacy in the Gambia for children aged 9 to 47 months using the given information. This calculation demonstrates that the measles vaccine in the Gambia has an efficacy of 89% for children aged 9 to 47 months. By comparing the attack rates between vaccinated and unvaccinated groups, we can determine the effectiveness of the vaccine in reducing the incidence of measles. The primary goal of the formula developed by Peter Chu is to simplify the calculation of vaccine efficiency. By breaking down the calculation process into easily understandable steps, individuals can grasp the concept more easily and engage with the information more effectively. It is crucial that we empower people to make their own assessments of vaccine efficacy. By providing them with a user-friendly tool, we encourage them to become active participants in their health decisions. This formula serves as a bridge, connecting scientific data with public understanding and facilitating informed choices. Ultimately, Peter Chu formula aim is to enhance vaccine acceptance. By demystifying the calculation process and offering a clear method for calculate vaccine efficiency, we can instill confidence in the public and promote higher vaccination rates. Let us empower individuals to make informed decisions and embrace the power of vaccination. Furthermore, the Peter Chu formula can assume a high target population of vaccination, such as the medical worker group, represented by a value of K equals 100. This assumption simplifies the calculation process even further, making it easier for individuals to grasp and apply. By simplifying a complex formula, the Peter Chu formula empowers individuals to engage in data-driven decision-making. It enables them to evaluate the efficiency of the COVID-19 vaccine and comprehend the impact it can have on their lives and communities. What sets the Peter Chu formula apart is its reliance on easily accessible data found in the news. It allows individuals to compare the average infection rates of both vaccinated and unvaccinated groups, enabling them to make informed assessments. This approach ensures that the formula remains transparent and accessible to all. The formula's simplicity aligns perfectly with Albert Einstein's famous quote, "Everything should be made as simple as possible." In addition, Albert Einstein's also quote, "One, we cannot solve our problems with the same thinking we used when we created them. 2, if you can't explain it simply, you don't understand it well enough. 3, genius is making complex ideas simple, not making simple ideas complex. 4, any intelligent fool can make things bigger and more complex. It takes a touch of genius and a lot of courage to move in the opposite direction. 5, God always takes the simplest way. 6, when the solution is simple, God is answering." Isaac Newton quote, "Nature is pleased with simplicity, and nature is no dummy." From the Albert Einstein's and Isaac Newton quote, it can be seen that simplifying knowledge is very important. For those interested in new simple knowledge, I recommend referring to my book titled Peter Chu Formula for Maximum Positive Rate Based on COVID-19 Mutant, 3rd edition. Simplifying Mechanical Engineering Solutions with Peter Chu Rule, Method and Theorem. Simplifying Electrical Engineering Solutions with Peter Chu Rule, Method and Theorem. And Simplifying Civil Engineering Solutions with Peter Chu Rule, Method and Theorem. The Peter Chu formula used to calculate the efficiency of the COVID-19 vaccine is similar to the sum of n terms and the sum of infinite terms geometric progression. In a geometric series, when the absolute value of the common ratio, r, is less than 1, raising it to the power of infinity will approach 0. Consequently, the sum of an infinite geometric series will converge to a value equal to the first term, a divided by 1 minus r. Similarly, in the Peter Chu formula, if we consider value k approaching 100, the vaccine efficiency can be calculated as b minus a, and then all divided by b and times 100%. Here, b represents unvaccinated infect rate, A represents vaccinated infect rate, and K% represents the percentage of a vaccinated group as compared with an unvaccinated group. By comparing these two formulas, we can observe the mathematical parallels between the convergence of a geometric series and the calculation of vaccine efficiency using the Peter-Chu formula. According to article from Proofs in Mathematical Reasoning, Agata Stefanowicz et al., 2014, mathematical proof is absolute, which means that once a theorem is proved, it is proved forever. Until proven, though, the statement is never accepted as a true one. Therefore, a mathematical proof of Peter-Chu's formula is provided to ensure its soundness. Now, let's take a look at an example from a study published in the New England Journal of Medicine, titled Safety and Efficacy of the BNT162b2 mRNA COVID-19 Vaccine by Fernando P. Polak et al. 2020. This study provides valuable insights into the safety and effectiveness of the vaccine. During the study, a total of 43,448 individuals received injections, with 21,720 receiving the BNT162b2 vaccine and 21,728 receiving a placebo. Among those who received the BNT162b2 vaccine, there were 8 cases of COVID-19, compared to 162 cases among those assigned to the placebo group. First, let's put the data to a tablet. This table provides a concise overview of the distribution of individuals and the respective number of COVID-19 cases in each group, highlighting the significant difference in the occurrence of COVID-19 between the vaccinated and placebo groups. To calculate the vaccine efficacy based on the provided data, we can use the original formula. Vaccine efficacy equals attack rate in unvaccinated group minus attack rate in vaccinated group, and then all divide attack rate in unvaccinated group and times 100%. Given that the attack rate in the placebo group is 162 cases out of 21,728 individuals, the attack rate can be calculated as attack rate in placebo group equals 162 divide 21,728 times 100% approximately equals 0.7459%. Similarly, the attack rate in the vaccinated group is 8 cases out of 21,720 individuals, leading to attack rate in vaccinated group equals 8 divide 21,720 times 100% approximately equals 0.0368%. Using these values, we can calculate the vaccine efficacy. Vaccine efficacy equals 0.7459% minus 0.0368%, and then all divide 0.7459% approximately equals 95.0599%. Therefore, based on the normal calculation using the given data, the vaccine efficacy is determined to be approximately 95.0599%. This indicates a significant reduction in the incidence of COVID-19 among the vaccinated group compared to the placebo group. Indeed, it appears that when we apply Peter-Chu's formula, we obtain the same result for vaccine efficacy as the normal calculation. Both methods yield a vaccine efficacy of approximately 95.0599%. This consistency reinforces the validity and reliability of Peter-Chu's formula in assessing vaccine efficacy. The formula provides a mathematical approach to determine the effectiveness of a vaccine based on the number of cases in the vaccinated and placebo groups. In this particular case, whether we use the normal calculation or Peter-Chu's formula, we arrive at the same conclusion that the BNT162b2 mRNA COVID-19 vaccine has a high efficacy rate of around 95.0599%. This signifies a substantial reduction in the incidence of COVID-19 among those who received the vaccine compared to the placebo group. It's worth noting that the agreement between the two calculations further supports the soundness and applicability of Peter-Chu's formula in evaluating vaccine efficacy. When we use Peter-Chu's formula with a value of K equals 100, the calculated vaccine efficacy is approximately 95.0617%. This value is indeed very close to the normal calculation result of 95.0599%. The negligible difference between the two calculations further reinforces the reliability and consistency of both methods in determining the vaccine efficacy. Both approaches lead to similar conclusions, indicating a high level of effectiveness of the BNT162b2 mRNA COVID-19 vaccine in reducing the incidence of COVID-19. It highlights the robustness of Peter Chu's formula and provides additional evidence for the reliability of both calculation methods. It is evident that all three calculations, normal calculation, Peter Chu's formula, and Peter Chu's formula with K approximately equal 100, yield nearly identical results for the vaccine efficacy. The fact that these calculations produce consistent results further strengthens the validity and reliability of the methods used to determine vaccine efficacy. It provides confidence in the accuracy of the assessments and reinforces the conclusion that the BNT162b2 mRNA COVID-19 vaccine has a high efficacy rate of approximately 95%. These aligned results from multiple calculation methods enhance the overall understanding and trustworthiness of the vaccine's effectiveness in preventing COVID-19. It underscores the importance of utilizing different approaches and methodologies to cross-validate findings, ensuring robustness and accuracy in scientific analysis. On November 18, 2020, Pfizer and BioNTech announced the completion of the Phase 3 study for their COVID-19 vaccine candidate, BNT162b2. The study results showed that the vaccine met all primary efficacy endpoints. The primary efficacy analysis revealed that BNT162b2 was 95% effective against COVID-19, starting 28 days after the first dose. In the study, a total of 170 confirmed cases of COVID-19 were evaluated. Out of these cases, 162 were observed in the placebo group, while only 8 cases were reported in the group that received the vaccine. These results are significant and demonstrate the vaccine's effectiveness in providing protection against COVID-19. The data indicates that receiving the BNT162b2 vaccine significantly reduces the risk of contracting the virus. Please note that this information is accurate as of November 18, 2020. If we consider a modification in the data where the vaccination process only reaches 90% of the total target group, we can adjust the vaccinated data accordingly. The original vaccinated data is 21,720 individuals. If we take 90% of this value, we would have 0.9 times 21,720 approximately equals 19,548. The original infect COVID-19 is 8 individuals. If we take 90% of this value, we would have 0.9 times 8 approximately equals 7.2. Even with the modification where the vaccination process reaches 90% of the total target group, the results from the 3 calculations remain almost the same. This further demonstrates the reliability, consistency, and robustness of Peter Chu's formula in assessing vaccine efficacy. Regardless of the vaccination coverage, whether it is 100% or 90%, the vaccine efficacy calculated using Peter Chu's formula remains highly. This consistency highlights the inherent strength of the formula in evaluating the effectiveness of a vaccine in reducing the incidence of a specific outcome, such as COVID-19 cases. The fact that the results from the different calculations align closely affirms the validity of Peter Chu's formula and its practicality in assessing vaccine efficacy. It provides reassurance to researchers, healthcare professionals, and the public, reinforcing confidence in the formula's application and the conclusions drawn from its results. It is noteworthy that Peter Chu's formula provides a straightforward and accessible method to evaluate vaccine efficacy, allowing for reliable comparisons and informed decision-making in public health interventions. To learn more about Peter Chu's formula for calculating COVID-19 vaccine efficiency, you can find articles on reputable platforms like the World Health Organization (WHO) or Europe PMC. Europe PMC provides access to the full paper of the article. For a comprehensive understanding of the topic, I recommend referring to my book titled Peter Chu Formula for Calculate COVID-19 Vaccine Efficiency, 3rd edition. This book will provide you with detailed information on the formula and its worksheet. In a study published by the Journal of Infectious Diseases titled Vaccine Epidemiology, Efficacy, Effectiveness, and the Translational Research Roadmap, Jeffrey A. Weinberg et al., 2010, an alternative and equivalent formulation of vaccine efficacy is presented. According to this formulation, vaccine efficacy VE can be calculated using the equation VE equals 1 minus RR times 100%. Here, RR represents the relative risk of developing the disease for vaccinated individuals compared to unvaccinated individuals. This alternative formulation provides a different perspective on calculating vaccine efficacy, emphasizing the comparison of relative risks between the vaccinated and unvaccinated groups. It serves as an additional approach to quantifying the effectiveness of vaccines in preventing the occurrence of diseases. The relative risk, RR, is the ratio of the probability of an outcome in an exposed group to the probability of an outcome in an unexposed group. For example, in a study examining the effect of the drug apixaban on the occurrence of thromboembolism, 8.8% of placebo-treated patients experienced the disease, whereas only 1.7% of patients treated with the drug experienced the disease. Therefore, the risk ratio is calculated as 1.7 divide 8.8, which is 0.19. This can be interpreted as those receiving apixaban had 19% the risk of recurrent thromboembolism than did patients receiving the placebo. In this case, apixaban is considered to be a protective factor rather than a risk factor because it is associated with causing a reduced risk of disease. Assuming the causal effect between the exposure and the outcome, values of RR can be interpreted as follows. RR equals 1 means that exposure does not affect the outcome. RR less than 1 means that the risk of the outcome is decreased by the exposure, which can be called a protective factor. RR greater than 1 means that the risk of the outcome is increased by the exposure. Now, let's shift our focus to how the public can calculate the efficacy of COVID-19 vaccines using information from local news sources. For instance, there was a calculation conducted for Pfizer-BioNTech's COVID-19 vaccine efficacy, specifically for Malaysian healthcare workers. The news article, titled COVID-19 Outbreak in Malaysia and published on January 25, 2020, reported the first case of COVID-19 detected in Malaysia. According to a news article dated February 5, 2021, it was reported that nearly 5,000 healthcare workers had been infected with COVID-19 so far, with 2/3 of them being women. Before the vaccination campaign began, a total of 4,756 healthcare workers had been infected. Now, let's calculate the average number of infections per day before the vaccination period. Assuming the start date as January 25, 2020, the day when the first case of COVID-19 was detected in Malaysia, and considering the timeframe until February 5, 2021, which is approximately 376 days, we can determine that the average number of healthcare workers infected per day before vaccination was 12.65. Moving on to the vaccination progress, according to news released on March 17th, the second dose of COVID-19 vaccinations was scheduled to begin. Based on the Centers for Disease Control and Prevention, CDC guidelines, full vaccination is considered achieved on April 1st, 2 weeks after the second dose. A subsequent news report on April 17th mentioned that 9 healthcare workers were infected after being fully vaccinated. Considering the time from April 1st to April 16th, which is 16 days, we can estimate that an average of 0.5625 healthcare workers were infected per day after being fully vaccinated. Note, the revised content has been further simplified and adjusted for clarity. It's worth noting that the calculation results align with a real-world study conducted on Pfizer-BioNTech's COVID-19 vaccine, which showed an effectiveness of approximately 95%. A ratio of 1 divided by the relative risk, RR, of 22.47 indicates that the risk of infection among the unvaccinated population is 22 times higher than that of the vaccinated population. According to a news report on February 5, 2021, a real-world study conducted among healthcare workers in Malaysia demonstrated that the Pfizer-BioNTech vaccine was 95% effective in preventing COVID-19. This finding aligns with the calculation results obtained using the Peter-Chu formula, further validating its accuracy. Peter-Chu formula calculation result also is consistent with the real-world study conducted by Pfizer-BioNTech's COVID-19 vaccine, and its effectiveness is about 95%. Referring to the table shown, if healthcare workers had not been vaccinated, it is estimated that approximately 202 infections would have occurred between April 1st and April 16th. This means that 202 healthcare workers would have been infected during that period. Additionally, considering the previous data, an additional 193 healthcare workers would have been infected prior to April 1st. These numbers are significant because infected medical staff can transmit the virus to vulnerable patients in hospitals. This transmission can result in potential complications and, in severe cases, even deaths among the patients. The importance of vaccination becomes evident when we consider the potential impact on both healthcare workers and the patients they care for. By reducing the number of infections through vaccination, we can protect not only the healthcare workers themselves, but also the wider community, including vulnerable individuals who depend on their care. These insights highlight the significance of widespread vaccination efforts and the role they play in mitigating the spread of the virus and preventing further harm. Considering that some members of the public may have limited computing power or lack the time to perform complex calculations, it would be beneficial to integrate the new Peter Chu formula for calculating COVID-19 vaccine efficiency into user-friendly tools like the PCET medical calculator. The PCET medical calculator can serve as a convenient and accessible platform for individuals to input relevant data and obtain the vaccine efficiency results without having to manually perform the calculations themselves. By incorporating the Peter Chu formula into the calculator, users can simply input the necessary parameters, such as the number of infections in vaccinated and unvaccinated groups, and obtain the vaccine efficiency output instantly. This approach reduces the burden on individuals to perform complex calculations manually, ensuring that vaccine efficiency can be easily understood and accessible to a broader audience. By leveraging technology and user-friendly tools like the PCET medical calculator, we can empower individuals to make informed decisions and better understand the effectiveness of COVID-19 vaccines without requiring advanced mathematical skills or extensive computational resources. On the COVID-19 related formula page, there will be two options available for calculating the vaccine efficiency. By selecting conventional formula option, users can calculate the COVID-19 vaccine efficiency using the conventional formula commonly used in vaccine evaluation. This formula may involve parameters such as the number of infections in the vaccinated and unvaccinated groups, and it provides a standard approach to assessing vaccine effectiveness. By selecting Peter Chu formula option, users can calculate the COVID-19 vaccine efficiency using the Peter Chu formula, which offers an alternative perspective on evaluating vaccine effectiveness. This formula considers parameters like the number of infections and provides insights based on the reduction in infections achieved by the vaccine. To learn more about vaccine efficiency and relative risk, users can click on the title, which will provide detailed information and explanations regarding these concepts. If users wish to exit the page or end the calculation process, they can click on the buy logo, which will conclude the session or game. By offering these options and providing information about vaccine efficiency and relative risk, the COVID-19 related formula page aims to facilitate understanding and enable users to choose the calculation method that best suits their needs. On the Peter Chu formula page for calculating the efficiency of the COVID-19 vaccine, there will be two options available. Option 1, K not equal to 100 users can choose this option to calculate the vaccine efficiency using the Peter Chu formula when K is not equal to 100. Users can input the relevant data, such as the value of K, the number of infections in the vaccinated and unvaccinated groups, and obtain the corresponding vaccine efficiency result using the formula. Option 2, K equal to 100. When users select this option, the calculation becomes simple, because without the need to calculate the value of K. To return to the home page or main menu of the PCET calculator, users can click on the PCET calculator logo button. This allows users to navigate back to the starting point or explore other functionalities offered by the calculator. On the normal calculation page, the following options and features are available. Calculate button. After inputting all the necessary data for the calculation, users need to press the calculate button twice to initiate the calculation process. Note, users should press the space where they want to input the number and then enter the corresponding value. By clicking on the title, users can access an example or sample calculation. This example provides a demonstration of how to input data and perform the calculation correctly, offering guidance and clarity for users. On the Peter Chu formula page for calculating the efficiency of the COVID-19 vaccine when K is not equal to 100, users will need to follow the instructions provided to ensure accurate input and calculation. Here are the steps. Key in the corresponding value for COVID-19 positive cases in the vaccinated and unvaccinated groups, and the value of K. Press calculate 2 times. After entering all the necessary data, users should press the calculate button twice to initiate the calculation process. These instructions and options are designed to ensure that users input the relevant COVID-19 positive case data accurately. Perform the calculation by double pressing the calculate button. Access examples for guidance, and easily return to the home page for further use of the calculator. On the Peter Chu formula page for calculating the efficiency of the COVID-19 vaccine when K is equal to 100, the calculation becomes simple, as there is no need to calculate the value of K. Users should enter the corresponding value for COVID-19 positive cases in the vaccinated and unvaccinated groups. There is no need to enter the value of K, since it is already assumed to be 100. Press Calculate 2 times. After entering all the necessary data, users should press the Calculate button twice to initiate the calculation process. Workshop Peter Chu Formula for Calculate Vaccine Efficiency by Southeast Asian Minister of Education Organization. Regional Central for Education in Science and Mathematics, or CMOA-REXAM. Theme: Education 4.0 Issues, Challenges, and Future Direction towards CMOA Priorities and Sustainable Development Goal (SDGs). Phase 2. Date: the 18th of May 2022. Venue: ICT Lab 1, CMOA-REXAM, Penang, Malaysia. Collaborating partner: CMOA Secretariat, Bangkok, Thailand. The workshop explored convention formula and Peter Chu's formula to calculate vaccine efficacy. Peter Chu's formula provides a simplified method that allows participants to easily calculate and understand vaccine effectiveness. Comment from a participant of workshop Peter Chu formula, Dr. Chin Chi Kiong.
Conventional formulas for calculating the efficacy of COVID-19 vaccines can be very complex. However, Peter Chu formula is so much easier. I agree that by using public news information, the public can easily calculate the efficacy of a COVID-19 vaccine using Peter Chu's formula. This is to ensure the public can calculate and understand the efficacy of the vaccine for themselves, thereby convincing the public to get vaccinated against COVID-19. I agree that simple formulas like Peter Chu formulas for calculating the efficacy of COVID-19 vaccines are important to help the vaccine industry, and recommend organizing more Peter Chu formula workshops for vaccine industry experts to learn how to use Peter Chu's formula for calculating COVID-19 vaccine efficacy. I prefer to calculate the efficacy of the COVID-19 vaccine using the PCET medical calculator rather than the normal calculator.
By encouraging vaccination and implementing effective preventive measures, we can work towards reducing the burden on healthcare systems and saving lives. Let us continue to prioritize vaccination and take necessary precautions to ensure the safety and well-being of everyone.
Thanks for listening.
Okay. Any question? Organizer? Hello.
Hello, yes, doctor.
Yeah.
Okay, thank you, doctor. Thank you for your presentation. I would like to ask the participants if they have any questions, they can ask. If any of you have questions, feel free to ask. In case if there are no questions, then we can move forward. And if there will be at the end of the session, we will be discussing, there will be a live discussion, then we can ask the remaining questions. Okay, thank you, doctor, once again for your presentation. Now I would like to
Thanks.
call upon our next keynote speaker, Dr. Dror Malka from Holon Institute of Technology, Israel. And he'll be presenting his presentation on the title Designing All Optical Logic Half-Ether with Photonic Crystal Multi-Range Resonators. Hello, Dr. Dror, are you there?
Thank you. Thank you for the nice introduction. I want to start.
Yeah, you're most welcome. You can start your presentation.
Let's share the screen.
Okay.
All right, I want to continue to share my screen. Let's find my presentation. Okay, let's do full screen. Okay, so everyone saw my presentation?
Yes, it is visible, doctor.
Yes, okay, let's start with the start. Okay, so my name is Dr. Dror Malka from Holon Institute of Technology. And today we're going to talk how to design half adder, a logic device, which is very important, especially for the topic for quantum computing and to realize a new field in photonics. So the topic is designing all optical logic half adder with photonic crystal multi-ring resonators. Okay, let's start. Start the movie, special effect. Okay, so what I'm going to talk about, what is optical logic devices, what is logic half adder, what's so special about this device and why it's so important to make it in optical components. Of course, the devices, the waveguide, the photonic crystal, the motivation of this work, why I'm doing this, the principle of work, the design, the new half adder technique, the logic design, the AND and XOR gate. After that, we're going to review the special effect of the photonic band diagram and the AND gate simulation results, the XOR gate simulation result, calculation of stability time, and in the end, optical transmission spectrum. I'm going to do comparison between this work to the other state-of-the-art work dealing with quantum computing and half adder devices. And finally, in the end, I'm going to conclude my talk. Okay, so let's continue. Okay, so optical logic devices component have attracted much attention due to their strong capability in optical communication field and potential to support computing technique. Usually in a special field like quantum computing, which is become very hot topic today. So the application, of course, is signal processing, quantum computing, and of course, to realizing optical arithmetic logic unit. Here we can see an example of the devices that we are trying to build, a photonic chip that maybe can integrate it in the future in computing a quantum unit. Okay, let's continue. Here is the very basic logic of adder. We can see the XOR and the AND gate, two input A and B in this example, and sum and carry is the output. We can see the truth table. So generally, the half adder consists of two logic gate, XOR and AND gate. The output of half adder sum and carry output are identical to the output of the logic gate. So this device mechanism of two input and two output, and it's basically can operate at functionality only for one bit with another bit. Okay, so if we want more things, we have to realize and create more devices. If, for example, if we want to create a full adder, we have to use two half adder with OR gate. And if we want a 4-bit adder, we need four devices of full adder to realize this combination of device. Okay, so let's continue. What is photonic crystal structure and what's so special about that? So photonic crystal is a unique structure based on the microstructure formation of high and low effective index materials, which is amazing because usually if I use optical fiber, I cannot use two materials with high and low index material. I have to use a very narrow space between the effective index value, which is because of the total reflection mechanism of the light guiding. So photonic crystal utilize the photonic band gap alongside to the modified total internal reflection mechanism, allowing design of new compact optical components, which is very unique property, and we're going to utilize it because it's better to use this waveguide because it's more compact and it can support semiconductor devices, and we don't want to fabricate it a scale footprint very large. Okay, now the motivation of this work. So the motivation, of course, from commercial view, of course, is money, because if we design a logic gate from photonic view, it will be much more faster, and also it will be much shorter and compact devices, which gain a lot of money from energy view, from cost of materials. So a variety of technology have been used to design optical logic gate, including waveguide, interferometer, optical fiber, semiconductor optical amplifiers. But this technology have a lot of limitations, such as low speed, very big size, which cannot be integrated with a laser. And also, if you want to integrate laser with these devices, there is a lot of problem with the mismatch of the modes. So much of the energy going to be lost, so a lot of losses. So in a chip scale level, it's very difficult to do that. This is why we use a photonic crystal. But we have another novelty that we are going to use photonic crystal without using the traditional nonlinear effect, without nonlinear materials. Usually, if we compare our work to other works, everybody are using with nonlinear materials, which is very difficult for using because nonlinear materials provide a lot of trouble of noise and nonlinear effect. Okay, so the principle of work of this guiding mechanism is to use a photonic crystal with a shape of ring resonator using a ring shape waveguide that is constructed from a photonic crystal with periodic modulation in the refractive index. This gives us the benefit of creating a band gap in the transmission spectrum of the waveguide and result in the confinement and resonance enhancement of light at specific frequency or wavelength. This is a special property that we're going to utilize. The phase difference in a photonic crystal ring resonator in our work, we do it as a race kite, can be controlled by adjusting the physical dimension of the resonator, such as the radius or the predicator of the photonic crystal, which is the pitch. Usually is the distance between two rolls. In our case, is the distance between two rod silicon. By optimize the phase difference, the resonance frequency of the photonic crystal ring resonator can be tuned. I can control it to achieve the operation wavelength of 1550 nanometer, which is the special wavelength of the C-band because of the low absorption in this range and also low power losses. Okay, so our new half-adder technique, a new structure for optical half-adder without non-linear material has been proposed. The proposed half-adder is built by a symmetrical configuration of all optical and XOR logic gate that are based on cross-shaped photonic crystal waveguide. Our specific aim in this project is to support the C-band, clear destination between level 1 logic and level 0 logic, which in our case is 1 logic going to be strong light and 0 logic going to be lower light level, low losses of course, and of course we want a compact device which will be very easily to integrate in CMOS technologic. Okay, so we're going to continue. Here is the design. We can see in this design that we have two splitters that have low losses. The red color in this figure represents the silicon red, and the white color represents silica. And we have XOR gate, and we have AND gate, and we have two input, A and B, and S and C are the output, like I showed you early in the logic half-adder design. Okay, so now we can see the AND and XOR logic design. So if we can see, if I start with the AND design, we can see the cross-shaped of 3 waveguiding. Here we see in the AND gate, and we have a racetrack here and here, 2 racetrack. The same principle here, we have 2 racetrack. Only we have 4 waveguide. In the horizontal, we have 3, and the vertical, we have 1, as we can see here. So this is the difference. So now we continue and see the result of the simulation. First of all, we simulated using plan waveguide expansion. We used to solve the photonic band diagram. After we optimize the pitch size, the pitch is the distance between the two silicon rod. And we optimize also the diameter of each silicon. After we optimize that and make a lot of simulation, we obtain these results. We can see the TTM band structure of the AND gate and the TTM band structure of the XOR gate, which show that we succeed to confinement the wavelength that we want. So the result showed that the wavelength range of the AND gate is between 1,470 nanometer, between 1,660 nanometers, and the wavelength range of the XOR gate is very similar. It's 1,510 nanometer to 1,920 nanometer. So the common wavelength range is support the C-band range. This is what we want. We want to enhancement this area that now we can confinement the photons inside this area. So if we use the operation wavelength in this area, in this range, we can enhance the light. So it's like using non-linear materials, but without using them. This is the novelty here. Okay, so here is the AND gate simulation results for state 0, 0 and 0, 1. We can see that we obtain in 0, 0 very low energy, and 0, 1 we have higher energy, and 1, 0 higher energy, and 1, 1 very high energy. And we conclude the results. We can see we have the results that we want to see. We want to see AND gate. So if we remember in AND gate, 0 and 0 will always give a 0. In our proposed work, we can see very low light, very low photons that collected in the output, and very strong output when we have 1. So this is what we want. So now we can continue. Here is the XOR gate simulation results for 0 and 0, the same principle of work. We see how the light is coupling. By the way, the light is coupling from the ring to the waveguide, and then again from the ring to the waveguide. This is why we optimize this all geometrical parameter to make this device to work in special mechanism and to enhancement the light. So there is not a lot of losses here in the coupling, and the photon is confined here in the ring because of the photonic band gaps, and then we have more energy getting outside. So this is why the XOR gate is working very good in 0, 1 and also in 1, 1, the same principle of guiding mechanism. And we conclude the results. Then after that, we calculation the imported stability time, which is very important parameter because we want to see that our photonic integrated circuit is very fast comparing to electronic devices or comparing to other photonics devices. So in our case, after we calculated the overall stability time, the stabilization time for the AND gate is only 0.66 picosecond, and the stabilization time for XOR gate is only 0.4 picosecond. So in our case, the overall stability time is going to be to the slower gate. In our case, is the AND gate, which is 0.66 picosecond. And after that, we also want to see that our devices have a strong stability for laser drift effect that can happen in real-time system, in a transceiver, or in when the laser is getting hit with the temperature is going to up, the laser is drift from its location. So we can see that we have a very good stability when we test the optical transmission spectrum for the half adder. It can be noticed that our proposed device can function well with a minor shift of 5 nanometers from the operated wavelength of 1,550 nanometers, and the half adder can handle the laser drift effect of the source that can happen because of the laser heating process, as I mentioned earlier. So we have a good result here. We can see the result for state, logic state, 0, 0, 0, 1, 1, 0, and 1, 1, and again also for the AND gate with different color, of course. Also, in the end, we're going to do comparison between this work to other work. So we can see our work is below. Here is the photonic crystal multi-ring resonator. So we can see that our work is very speed comparing to others. Also, others are much more speed, but we have a better distinguish of the logic state. Here they take from 0 to 0.35. You can see in our case it's 0 to 0.2 and 0.8 to 1, the logic state for 1. So it's very clear where is 1 and where is 0. This is reducing all the system error that can happen in the map system when you want to distinguish between 0 and 1 in the system operations. And also the size is very small, which is what I wanted in this work. Okay, so I want to conclude my talk. So a novel design for all optical and an XOR gate combined into an all optical half adder using a special configuration based on photonic crystal ring resonator in racetrack structure. By the way, the benefit that by using racetrack is that the coupling light is better, but the trade-off is that it's take more time to the photon to get out. Two-dimension square lattice structure without nonlinear material is presented. Results show that the intensity at light logic gate output showing that the logic state 0 range of the whole optical adder is between 0 to 0.3 normalized intensity, and the logic state 1 range is between 0.8 to 1, and the overall stability time is only 0.66 picosecond, which is an excellent result for us. And the advantage of this proposed half-adder design is clear distinction between logic 1 and logic 0 with a narrow power range that can minimize the logic error at the photonic decision circuit. This is a huge advance for this technology, very huge advance that don't have to other components that we investigate and try to find. And also this new half-adder can be used in quantum computing for photonic quantum computing calculation. These are our next work. Our next project is to, of course, is to take two half-adder with a OR gate and to do a photonic circuit, photonic intergate circuit, and to realize a full adder. And then from there, we are going to take this full adder and try to realize a four unit full adder. And then eventually, in the end, we want to create a photonic logic arithmetic unit process. Okay, so I want to thank to everyone that listened to me in this nice conference, this nice web, and thank you for your attention.
Thank you, Dr. Dror, for your presentation. And I would like to ask the participants if they have any questions, they can go on and ask. Okay, in that case, if there are no questions, we can move forward to our next presentation. Thank you once again, Dr.
Okay, thank you all.
Okay, now I'd like to call upon Dr. Carla Seri from University of Sassari, Italy, to deliver her speech on routine-loaded hybrid nanoparticles for nasal administration to protect from anthracycline-induced endothelial brain damages. Hello, Dr. Carla, can you hear me? A very warm welcome to you, Doctor.
Do you see my presentation?
Yeah, we can see your presentation and your video as well.
Sorry, I problem. Okay, so good morning, everyone. Thank you for introduction my work. I'm Carla Serri and assistant professor from University of Sassari. And my focus research about the production and preparation nanoparticle use different materials, polymeric and phospholipid material, and do you study the drug delivery. In this time, about focus about the intranasal deliveries on nose to brain. Today, my talk by title Routine Loaded Hybrid Nanoparticle for Nasal Administration to Protect from Anthracycline Induced Endothelial Brain Damages. The anthracycline is a chemotherapy standard anti-cancer drug composed of the doxorubicin, epirubicin, irarubicin, are currently used for the treatment of the different cancer, breast cancer, in the clinic in patient. The long treatment about the anthracycline do induce the collateral effect, radical damages, in particular acute and chronic inflammation, endothelial inflammation, vascular and cardiotoxicity, and brain damages. The ruti is a vegetable substance, is a citrus flavonoid glycoside, which is a low molecular weight polyphenolic compound. The rutin and quercetin is the flavonoid. It's very interesting they use this natural substance because have the properties anti-inflammatory, scavenger, and antioxidant. In particular, do you report in literature the benefits about the rutin, antibacterial, antiviral, potential cardioproperties, neuroproperties, and neuroproperties active substance? And do you see in the also by enhancing synapse plastic? The pharmaceutical properties of rutin is severely restrictive. It's by low water solubility, short half-life, and extremely poor viability, poor absorption in the small intestinal and metabolic intestinal bacteria on fever when do use administration by oral administration. For bypassar the problematic, the oral administration is very interesting, use the intranasal administration. In general, the intranasal administration, the nose is an anti-administration site that offers different advantages, like rapid and easy absorption in the systematic circulation, the first pass metabolism, as well as the direct brain targeting of drugs. In the context, this work, I propose the formulation, the hybrid nanoparticle for nasal delivery of rutin. In particular, I production the hybrid nanoparticle use the phosphatidylcholine, cholesterol, polosammeron, and hyaluronic acid. Then hybrid nanoparticle is characterized and study the chemistry and physical properties. Do you study the encapsulation of the rutin in the hybrid nanoparticle? And in collaboration from the Pascale from Naples, Italy, do you study in endothelial cells the cytotoxicity and anti-inflammatory and vascular properties effect of rutin? In particular, for the production this hybrid nanoparticle, I use the different material, phosphatidylcholine, cholesterol, poloxameron, and hyaluronic acid. The phosphatidylcholine and cholesterol is a phospholipid used in general for the repair liposome. But the phosphatidylcholine is very interesting because do you use for the protection and repair for the biological membrane. The poloxamer is copolymer to block and report in literature the interaction about the phosphatidylcholine and hyaluronic acid. Hyaluronic acid is a very attractive natural polymer ligand of inflammatory tumor targeting because do you interaction about the CD44 and RAM receptor are overexpressed on the numerous type of solid tumor and inflammatory cells. In particular, the hyaluronic acid is biocompatible, non-immunogenic, and do you increase the loading the different drug. In this context, it's very interesting in the intranasal delivery for the mucosal adhesive properties. The nanoparticle, do you prepare about the modified nanoprecipitation technique? Do you use two-phase or phase compound composed about the phosphatidylcholine, cholesterol, and poloxamer? And do you add in the water phase composed about the hyaluronic acid and the poloxamer solution? Do you sonicate about 2 minutes? And do you obtain the hybrid nanoparticles uniloading routing and hybrid nanoparticles loading routing? The nanoparticles, do you characterize for study the physical properties about the dimensionality or size, zeta potential, and morphology? Do you test the dimensionally, use the particles control size, and do you obtain for the hybrid nanoparticles uniloading routine the size about the 117 nanometer? When do you loading the routine in the hybrid nanoparticles, the size do you increase about 30 nanometer? Do you obtain the negatively zeta potential and report in literature is the result hypothesization for the add the hyaluronic cases in the formulation, identical result for the increase the polydispersity index, and the polydispersity index values obtained indicate a heterogeneous population. That's in general, do you increase because do you use the each hyaluronic cases molecular aware and obtain the viscosity solution for the dispersion of the nanoparticle. The 10 imaging confirm the formation of the three-dimensional network. In particular, do you form of regular parallel trend arranged in almost spherical structure? It's possible the precision of on the filament in the imaging the particles at them. It's possible the three-dimensional structure, and in the surface is the coating hyaluronic cases. Do you study for the stability the hybrid nanoparticles in terms of the sites in water for 30 days, and in RPMI serum used for the cellular study? Do you obtaining the stability sites the particles in water? And in RPMI, it is very nice, very good stability, because I don't have the interaction with the proteins that set the medium serum. In continuous, do you study the thermal analysis for the test the free routine uniloading hybrid nanoparticles, hybrid nanoparticles loading routine for the different scanning calorimetric by the study, the interaction from the routine or encapsulation the routine in the hybrid nanoparticles. The result report the peak, endothermal peak about the routine loading hybrid, uniloading hybrid nanoparticles about the 120 degree, and the peak endothermal peak about the free routine about the 118 degree. When it's very interesting thing, where do you test the hybrid nanoparticle loaded routine, because I don't present the endothermal peak, the free routine. This result do you confirm the encapsulation of routine in the nanoparticles. After study the total drug in dispersion, do you obtaining the 70% the routine encapsulation in the dispersion, the solution, and do you study the permeation of the free routine and the hybrid nanoparticle loading routine. In particular, in this context, do you use two different membranes for the valuation, the affinity in vivo for the nasal mucosa? Do you use the hydrophilic membrane? And for do you study the simulator the hydrophobic membrane for the nasal epithelia? Do you report the result, the hybrid nanoparticles loading routine about the hydrophilic membrane? Do you release in the acceptor the routine about 15 minutes? When do you hybrid testing hybrid nanoparticles in hydrophobic membrane? do you begin the release about the 30 minutes. In general, it's possible obtaining the identical release about 18% the routine from hybrid nanoparticles at 120 minutes. All the 3 hour, all the routine do you release about the whole formulation. In collaboration with Pascale from Naples, do you study the cell viability and release LDH and of endothelial cells? And in particular, do you study the incubation in endothelial cells, the along epirubicin, epirubicin in combination with uniloading hybrid nanoparticle, loading hybrid nanoparticles, loading routing. The epirubicin do you induce in the endothelial cells the cardiotoxicity. And don't change the result when do you incubation in combination the epirubicin uniloading hybrid nanoparticles. But when do you test the epirubicin and free routing or hybrid nanoparticles loaded routing, do you obtain the crash, the cardiotoxicity? This result is hypothesization the properties vasculoprotective of the routing in the endothelial cells. Finally, do you test the activity, the anti-inflammatory test for valuation the expression of the cytokine and the interleukin. In general, the result obtaining the good the crash, the inflammatory increase the inflammatory effect, the hybrid nanoparticles in routine associated at the epirubicin. In conclusion, in this work, I propose the new method use the modified precipitation technique for preparation the hybrid nanoparticles. Do you study, do you obtain the stability sites in water and in RPMI? In vitro permeation study showed a sustained amount of routing from hybrid nanoparticle loaded routing of up to 120 minutes compared to free routing. And the cellular study indicate an anti-inflammatory phenotype by induced by hybrid nanoparticles loaded routing in cellular models. The future of development study, it's possible to prepare the hybrid nanoparticles with nanoprecipitation technique using the syringe pump and the test the hybrid nanoparticle study in vivo. Thank you for attention.
Hello, doctor. Thank you for presenting. If any of the audience have any questions, they can go on and ask. If not, then we can move on to the next speaker. Okay, doctor, in case there are no questions, I would like to move on with our next presentation. Thank you very much for joining us today.
Thank you.
Okay. Now I would like to call upon Dr. Radwan Al-Mufti from Tata Consulting Solutions, Canada, and he'll be presenting a speech on oral thin film. Hello, doctor. Are you there? Can you hear me? Hello, I think the doctor has not joined yet, so I would like to call upon next Dr. Vishakha Jaiswal. Are you there? Can you hear me? Hello, Dr. Vishakha. Can you hear me? Am I audible? Hello.
Hello.
Yes, Dr. Vishakha. Hi.
Yes, ma'am, I'm there.
Yeah, hi, most welcome. Will you be able to present now? Are you ready with your presentation now?
Can you give me 5 minutes?
Yeah, sure. Take your time. No problem.
Okay, thank you so much. Ma'am, my presentation is visible.
Yes, doctor, it is visible. We can see your slides.
Hello,
Yes, doctor.
I'm audible now.
Yeah, you're audible.
Hello.
Yes, doctor.
I'm audible now.
Yeah, you're audible.
Okay. Shall I start now?
Yeah, sure.
A very good afternoon to all. Greetings of the day from my side. Dear organizers, first of all, I would like to thank for giving me this opportunity to speak about my research work. Actually, I'm associated with, currently I'm associated with Amity Institute of Pharmacy, Lucknow campus, Amity University, Uttar Pradesh, Noida, and working as an assistant professor here. So my research work is on biopolymers. Recently, biopolymers are widely used in various drug delivery platforms. I have explored Nilambo nucifera, which is a commonly grown aquatic plant and widely available in the Asian countries like India, China. And I have explored its biopolymer potential for drug delivery. So why we are focusing on the biopolymers? Why not the synthetic polymers? We are known that in various drug delivery platforms, we have to use various polymers for drug release modifiers, suspending agent, carrier, adjuvants, and also as emulsifiers. So, but in spite of the using synthetic polymers, we are exploring biopolymers because they are widely acceptable or widely available in the nature. Our human body is also composed of natural polymers such as proteins, nucleic acids, various cellulosic components, starch, cellulose, lignin, chitin, which we are using. They are all of natural sources, various natural sources. The first advantage of these natural biopolymers are that they are biocompatible and biodegradable in nature. So their biodegradable nature makes them more useful or more convenient to use in drug delivery platforms, such in case as like we are using formulating any implantable drug delivery. So in that case, we don't have to worry that we have to take out the drug delivery platform or the drug delivery system from the body and to perform the surgery. In case of biodegradable polymers, we do not have to perform the surgeries on the patient. So biodegradable, one aspect of the biodegradable polymer is that, and the another aspect, it does not create any environmental pollution that like the disposal of their biopolymer. We don't have to think that of. It can be easily disposable in the atmosphere, which is already available. Next is the moving to the classification of biopolymers. There are various biopolymers available. First, the classification is based on the biodegradability, that are biodegradable and non-biodegradable, which the synthetic ones are mostly non-biodegradable, but some are like PLGA, PGA, they are PLA are biodegradable. Next is the classification based on the repeating unit. It may be composed of polysaccharides or saccharides, proteins, and nucleic acid. Next, based on the chemical composition, we can classify them as polyesters, polysaccharides, polycarbonates, polyamides, and vinyl polymers. Next is some specific features of biopolymers. Why biopolymers are special, what their physical or chemical properties are special, or what the advantages we get in the biopolymer. If first we discuss the physical properties, that are flexibility, viscosity, increased melting and boiling point, like we get more viscosity in different grades of the biopolymers. Flexibility if we are formulating any transdermal formulation. In case of transdermal formulation, we also use mechanical features also. We require mechanical features like shear strength, elasticity. Next is the thermal properties. As we are knowing that we are not only using the biopolymers in drug delivery, we are using in various fields of the biomedical sciences. The degree of crystallinity, stability of the biopolymer is also a very important aspect. Their conductivity. Next, the optical property, they are optically active or inactive, that can be used in other fields also. Next, various applications of biopolymers, like I have discussed, not only in pharmaceutical field, we can use it in pharmacy, medicine, food, agriculture, cosmetic sciences. Like in pharmacy, we can use for controlled drug delivery, we can formulate microcapsules, hydrogels, aerogels, liposomes, micro nanospheres, nanoparticles. If we talk about agriculture, we can use them as sustainable agro practices for water recovery and soil conditioning. We can use them in a delivery of agrochemicals, biosolvents, super solvents. Next, we can also explore them in the field of cosmetics. Like in cosmetics, we can use hydrogels. Hydrogels are basically also formulated for any kind of formulations for oral care, skin care, hair care, and like very popular brand L'Oreal is having using the nano formulation in case of cosmetic science. Next is the food industry. In food industry, we can formulate in case of packaging material, we can explore the biopolymers for as a food emulsifier in the coating, in the for packed foods, we can use the biopolymers. In case of medical application, we can use as implants like stunts for formulation of instant. We have drug-eluting stunts that are used for drug release into the heart, artery walls specifically. And for that, we can use polymers to modify the release for the skin tissue, for biosensing, and for the diagnostic purpose also. Next, biopolymers in the drug delivery. How the biopolymers can be incorporated in case of drug delivery. The biopolymers can be incorporated along with the filler materials, and then it can be converted into a formulation which can deliver our drug to our target site, or it can be bioactivity activation, or it can be used in the tissue engineering also. Next, coming to my research is explored. I have explored the Nilambo nucifera for its biopolymeric potential and its various application in the pharmaceutical field. Nilambo nucifera is an aquatic plant. Generally, it is in India. It is known as Indian lotus, lotus root, sacred lotus, beans of India. So it is belonging to the family of Nilambonaceae. It is having two species. I have explored nucifera species. So the plant rhizome are edible in nature. It is aquatic, as I earlier told. It is aquatic medicinal plant. It is having various medicinal properties like antidiarrheal activity, psychopharmacological, diuretic, antipyretic, antifungal, anti-yeast activities, hypoglycemic, hypotension, and hypercholesteremic effects it is having. And it is rich in dietary fiber, vitamin C, potassium, thiamine, various essential vitamins, and some minerals like phosphorus, copper, manganese, and having very low level of fat. So it is, as per dietary aspect, it is very good in nature, and it contains two alkaloids, that is luciferin and apomorphine. When the alcoholic particularly ethanolic extract was prepared of the Nilambo nucifera, it was containing glucose, fructose, and sucrose. And in case of cold water extract, and in case of ethanolic, 80% ethanolic extract, we got mannitol, galacturonic acid, and raffinol. This was the basis of my research, and the polysaccharide fraction was with 90% formic acid. We got the polysaccharide fraction, which was only containing the glucose. Next is the I have explored Nilambo nucifera as a biopolymer, but the platform or the formulation which I have formulated within Nilambo nucifera biopolymer was the nano emulsion. The nano emulsion is a fusion of nano emulsion, and it is a hydrogel where the physical state of drug containing the nano emulsion is changed by adding the gel matrix, thus enabling the more lipophilic drugs can be used in the treatment therapies. It resolves the major issues such as limiting use of lipophilic drug, poor bioavailability, unpredictable pharmacokinetic and absorption variation. Simultaneously, it is non-greasy because it is having hydrogel base, so it is non-greasy, easily spreadable, and better patient compliances. There are some several advantages of hydrogels like better skin permeability, better loading capacity, better stability, incorporate a lipophilic drug can be incorporated in the nanogels. It is having better patient compliances, better pharmacodynamic activity, pharmacokinetic profile like ADME. We got the better ADME. The response we get from the patient, therapeutic response is also better from in case of nano emulsions, controlled release, we get the better safety and better stability profile, and also the better drug loading, like higher amount of drug can be loaded into the nanomulsions. Next, how the isolation was performed. For the isolation, I have used the simplified procedure. In this, 250 grams of lotus roots were taken, and its outer portion was peeled out, and it was cut into the small pieces. Then it was grinded with water for 15 minutes in a mixer. Then I got a slurry which was filtered, and the supernatant was centrifuged, and the supernatant was treated with the optimized quantity of ethanol, and it was kept for refrigeration for 12 hours. Then it was centrifuged again, and the supernatant was collected. The supernatant was purified by hot dialysis method. After performing the hot dialysis, the biomaterial was recovered, and it was dried in a desiccator for 10 hours. Then it was passed through sieve number 120, and then it was stored for further use. The percentage yield what I got was 1.2% plus minus 0.04% standard deviation. Next, the characterization of Nilambo nucifera biopolymer was done. In the characterization, the physical-chemical properties. First, the physical properties, it was solid in nature, having pale brownish in color. It was odorless and bitter in taste. The melting point, which was observed by this for this biopolymer, was 200 to 210 degrees Celsius, which is quite high, and it can be easily incorporated in various formulations. Next, the biopolymer confirmed the chemical test of starch and protein, and it was devoid of protein. I think now my slides are moving or not. Shall I continue?
Your slide cannot see.
Hello, my slides are moving or not?
No, not see your slide.
My slides are visible.
No. Yeah, disappear.
It may be due to internet connectivity. Is it visible? Now is it visible?
Now it's okay.
Okay.
Yeah. Now okay.
Okay. Thank you so much. So in characterization, the physical-chemical characterization was done. The biopolymer confirmed the chemical test of starch, carbohydrate, and it was devoid of protein. The biopolymer was soluble in methanol and ethanol in distilled water, and it was insoluble in acetone. The various, I have performed the IR spectroscopy of this polymer. It showed peak of at 3,472 wave number. Then various peaks. Majorly the peak observed was of hydroxyl group, carboxyl group, and sulfhydryl group, which are responsible for the mucoadhesion properties of the biopolymer. Next, the composition of different batches of different formulation which I have prepared. The model press which I have taken was aripiprazole. It was in 10 mg in quantity. Next, I have taken carbopol as a standard polymer to compare the bipolymeric nature of the Nilambo nucifera. It was compared with carbopol 940, liquid paraffin, 280, span 20, propylene glycol, ethanol, and methyl and ethyl paraben was taken as a preservative. In the formulation number F1 and F2, I have don't taken Nilambu nucifera as a biopolymer, and F3 and F4 was formulated with the Nilambu nucifera biopolymer. F1, F2 was containing carbopol as a gelling agent. Next, the formulation aspect, the carbopol 940 was used as a gelling agent, and it was used to formulate a hydrogel. In first phase, the aqueous phase was prepared by dissolving the drug and the biopolymer. Next, in second phase, the oil phase was prepared, like dissolving the span, tween, and other ingredients. Then it was kept for continuous agitation. The hydrogel, which was I have prepared with carbopol 940, it was stabilized with triethanolamine and pH was adjusted to 6 to 6.5. And then the oil phase and aqueous phase was mixed to form a emulsion. Then it was kept for after mixing all the aqueous and oily phase, it was kept for continuous agitation. In this step number 1, we can see, and which will give our nano emulsion. After formulating a nano emulsion, the nano emulsion was mixed with the gelling agent, or that is hydrogel containing carbopol 940. And then the nano emulsion and gelling agent both were mixed to formulate a nano emulsion. The nano emulsion was formulated by mixing 1:1 ratio of emulsion as well as hydrogen. The next is the evaluation parameters which I have performed for nanoemulsions is for physical appearance, rheology. Rheology was performed by Brookfield viscometer, spreading coefficient, skin irritation studies, biodensity strength, in vitro release study, and stability studies were done for a period of 3 months. Later on, it will be extended for 6 months. Next, formulation 1, F1, F2, F3, F4. These are the results of evaluation parameters. The carbopol formulation was white in color, while the F3 and F4 was pale yellow to yellow in color. Homogeneity was excellent for both the formulation, for all the formulation. Consistency was also for very good, and no phase separation was visible in the all the formulations. Next, viscosity. In case of F2 formulation, I received the highest viscosity, while the viscosity of F3 and F4 formulations was lesser than the carbopol formulation. In case of spreading coefficient, spreading was comparable to the carbopol formulations, and the adhesion strength was also comparable. While in formulation F4, I got a little bit lesser adhesion strength, but it was comparable to the carbopol. Next, the release studies were performed using the France diffusion cell. In France diffusion cell, phosphate buffer pH 7.4 was used as a buffer medium to get the release, and the temperature was maintained 37 degrees Celsius. The order of the release was highest received for the formulation 4, then F1, then F2, and F3. The amount of that release highest was for F4. All the emulsions formulations were found to be stable for storage of 3 months, and no changes were observed in the physical appearance, pH, rheological properties, and drug content. This one, my paper is already published in material to the conference proceeding on the Nilambu nucifera. For the application, its biopolymeric potential, application of biopolymeric potential for site-specific delivery. These are my some references. And thank you so much.
Thank you, Dr. Vishaka, for your presentation.
Thank you so
Glad
much.
all the participants have gained critical insights from that. And I would like to ask if any of the participants have any questions for you. Please feel free to ask the questions. In case there are no questions, we would like to move forward to our next presentation. Thank you once again, Dr. Vishaka.
Thank you so
And
much.
if there are any questions from the participants, we can discuss it at the end of the end of the presentations of all of the speakers and posters.
Okay, thank you, sir.
Yeah, thank you.
Surely, sure.
Now I would like to propose a short break of 5 minutes. I request you all to stay in the meeting. Please don't leave the meeting room, and we can join after 5 minutes and continue with the presentations. Thank you.
हेलो हाँ जी सर जी जी सर अच्छा ठीक है ठीक है
Hello, can you hear me? Hello,
Hello.
hello.
Yeah, hi, Professor.
Live.
Lee. Actually, we are having a short break of 5 minutes. You can start your presentation after 5 minutes.
Okay.
Yeah. Just 5 minutes. Okay, thank you.
Yeah.
Yes. Yes.
Hello, Dr.
Hello.
Yeah, Professor Einstein, are you ready?
Thanks, Dutch.
Yeah, let me introduce you. As uh, as per this schedule, we are not going. I apologize as some of the speakers have still not joined. And thank you for joining us and cooperating with us, Professor Zhu.
Okay, thank you for introduce me.
Yeah.
Let's
Yeah.
begin. Hello, everyone. Good morning, good afternoon. My name is Jinfang Zhu from Xinjiang Agricultural University, China. My topic is enhanced anti-tumor efficacy and attenuated cardiotoxicity of dogs in combination with lectin lipsums. This paper was published in Journal of Lipsum Research. I would like to report the following 5 content: background and significance of research. Lectin is a natural red pigment frequently found in ripe tomatoes, fruits. As a natural antioxidant, it could be used for the prevention and treatment of cancer, cardiovascular disease. However, poor stability in solubility in water and low bioavailability limit the clinical application of lectin. DOX is a broad-spectrum anti-tumor agent which intercalates into the DNA and inhibits macromolecular synthesis. However, there is a treatment contradiction between anti-tumor and toxicity, especially irreversible cardiotoxicity. The mechanism is relevant to the oxidative stress. Therefore, a combination of some antioxidants such as lectin could effectively alleviate the DOX-induced cardiotoxicity. This is a technology roadmap. Firstly, LVC were prepared by a thin film hydration method to overcome the instability, insolubility, and low bioavailability of lectin. Their diameter, LCEE, and drug release in vitro were measured. Then in vitro cytotoxicity and in vivo anti-tumor activity were determined to evaluate the effect of LLYC and their combination with DOX. Finally, we evaluate whether LLYC could decrease the DOX-induced cardiotoxicity in vivo. The drug loading capacity and encapsulating efficiency of the lipsums determined by HPLC were 1% and 82%. The particle size of LLYC appeared uniform, and the average diameter was 160 nanometer with a PDI of 0.273. The LLYC solution showed red opalescence. TEM examination further confirmed that LLYC had a spherical vascular-like structure with a phospholipid bilayer. About 50% and 82% of the total lecpin released at pH 5.5 and 7.4 within 24 hours. pH-dependent release showed by LLYC may be due to the instability of lecpin and weak acidic condition. DOX inhibit the growth of B16 and HeLa cells by 44% and 39%. LYC inhibited the cell growth by 41% and 34%. When these cells were treated with the combination of DOX and LYC, the inhibition ratios of B16 and HeLa cells were further increased by 71% and 64%. The growth inhibitory effects of the combination on B16 and HeLa cells were both greater than those of the DOX treatment alone. This result suggested a synergistic inhibitory effect of DOX and LLYC on cell growth. The in vivo anti-tumor efficacy was evaluated in a murine melanoma model, as shown figure. Tumors grow rapidly in the mice treated with normal saline and reached volume of 1,251 cubic millimeter in 10 days, while the mean tumor volumes were only 291 433 and only 63 cubic millimeter in the groups administered DOX, LLYC, and DOX plus LLYC. Moreover, tumor grow even more slowly in the mice treated with the combination of DOX and LLYC. Oh, sorry. The anti-tumor effect of various treatments was further assisted by histological examination. The tumor sections from the control group showed a normal oval and intact architecture. In contrast, the tumor section from the DOX treated mice showed focal necrosis tumor cells with blood boundaries and vacuolization. Meanwhile, the tumor sections from the LYC treated mice showed focal necrosis, blood boundaries, and significant atrophy of tumor cells. Notably, the tumor sections from the mice treated with the combination of DOX and LLYC showed focal necrosis, blood boundaries, atrophy, and vacuolization of tumor cells. This result indicated that apoptosis occurred in B16 melanoma tumors treated with DOX LLYC and DOX plus LLYC, which was consistent with the results of tumor growth inhibitors. DOX is a highly effectively anti-tumor drug. However, the clinical use of DOX is limited because of its dose-dependent cardiotoxicity. Oxidative stress is its key mechanism. Therefore, lectin as a natural antioxidant may elevate the DOX inductus cardiotoxicity. As shown in figure, no noticeable difference in the body weight was observed between the mice treated with LYC and the normal health, this line and this line. However, the body weight of DOX treated, this is DOX treated mice was significantly lower. His body weight is lower. To determine the extent of cardiac injury, histopathological examination of heart tissue was carried out. The heart sections from the normal control showed a normal striated cell distribution and a normal myocardial architecture. In contrast, the heart sections from the DOX-treated mice showed focus necrosis, disorganization, and focal fragmentation. Intramuscular infiltration of inflammatory cells and cell vacuolization. Treatment with LLYC preserved the microcardial architecture and showed no histopathologic changes relative to the normal healthy mice. This result indicates that LLYC might attenuate DOX-induced cardiotoxicity. During clinical follow-ups, CK and LDH are measured as a diagnosis mechanism of myocardial infarction. In this study, DOX-induced cardiac damage was clearly indicated by increased in plasma activity of CK and LDH, which could be ascribed to the excessive production of free radical and lipid peroxides, result in cell membrane damage and consequent leakage of this enzyme from heart tissues. Treatment with LYC result in a significant reduction in the plasma CK and LDH levels. Meanwhile, the DEX induced redox in imbalance in heart tissues was assessed by determining cardiac or oxidative stress biomarkers such as MDA and GSH. Administration of LLYC significantly attenuate the MDA level and remarkably elevate the GSH level compared with those in the DOX group. Conclusion. This study focused on enhancing the anti-tumor efficacy and attenuating the cardiotoxicity of DOX in combination with LLYC. First, LLYC were constructed with the aim to overcome the instability, insolubility, and low bioavailability of lectin. The particle size of LYC appears uniform. Compared to DOX treatment alone, a combination of LYC and DOX produced a significant increase in cytotoxicity in vitro and decreased the tumor size in B16 melanoma bearing mice in vivo. Additionally, the administration of LLYC significantly attenuates the MDA levels and remarkably elevated the GSH levels compared with those in the DOX group. Furthermore, treatment with LLYC resulted in a significant reduction of CK and LDH levels compared with those in the DOX groups. Those results were confirmed by histopathological examination. Overall, LYC could attenuate the DOX-indexed cardiotoxicity while their antioxidant properties and to enhance DOX anti-tumor effects. Thus, a combination of LYC and DOX may be a promising regimen to be used in clinical practice for patients on the DOX chemotherapy. Acknowledgement. This study was financially supported by National Natural Science Foundation of China and Xinjiang National Science Foundation. Thank you for attention. That's all. Thank you. Is there some question? Is there any question?
Thank you, Dr. Wu, for your presentation. Yes, if the participants have any questions, they can join and ask. In case there are no presentations, I would like to move forward to our next presentation. Thank you once again. And if the participants have any questions, we can discuss it at the end of this discussion.
Thank you.
Thank you so much. Now I'd like to call upon our next speaker. Mr. Siko Jose from Mahatma Gandhi University, India, and he'll be presenting his speech on green composites prepared from sheep wool reinforced vulcanized rubber with enhanced physical and mechanical properties. Hello, doctor. Can you listen to me?
Yeah, thank you.
Yeah, you can.
Thank you very much, the organizers, for giving me an opportunity present my research finding in the conference. Am I audible?
Yeah, you're audible.
And my slides are visible?
Actually, we can't see it now. There is a blank space. Try sharing it once again.
I kept it in slideshow mode. Is it moving? Slides are moving?
Yeah, now we can see it.
Okay, fine. Thank you. So today I would like to discuss some development of composites using natural rubber with the sheep wool. Myself, Siko Jose. I am from Textile Manufacturing and Textile Chemistry Division, Indian Council for Agriculture Research. And along with me, my teammates, Mr. Shanuman, Anmi Paul, Adityan, and Professor Sabu Thomas. Now, before going to the topic, I just would like to give a brief introduction about the non-biodegradable materials such as plastics and their harmful effects on humans and ecology. All of you know that plastics are not biodegradable, but still it is an essential part of our life. We cannot imagine a world without plastics. So an exact replacement of plastic is not possible. And what we are doing to do is we are trying to reduce the consumption of plastics, or we are trying to recycle the plastics. Now, there are other things, other alternatives are there, which can be a partial replacement of plastics or non-biodegradable materials, especially which are coming from natural fibers and natural fiber composites. So this slide is a small representation or examples of some of the materials which are coming from natural fiber reinforced polymer composites, which are either it is 100% biodegradable or it might be partially biodegradable. That depends, the degradability of the material or the composite depends upon the type of the type of polymer which we are using. Suppose if we are using a polymer which is biodegradable and a fiber, natural fiber, which is also biodegradable, obvious. So we ultimately we can go for a biodegradable composites. But in all cases, it may not be possible. So in certain cases, we will prefer natural fiber and synthetic resins, just like epoxy, unsaturated polyester resin and everything. It's already available. It is already established in the market. It is already established in some of the industries. So let's see how we can work on a fiber reinforced composite with the natural rubber. So my work is on natural rubber and wool polymer composites. Wool is a fiber, it's a sustainable fiber obtained from sheep. Natural rubber is also taken from a tree. So I am trying to combine these both natural fiber as well as rubber through mastication. Initially, the rubber has to be masticated. After that, we will add some vulcanizing chemicals into that, followed by the addition of wool fiber. Now, this process is done in two-roll milling mixing. After that, the mixture, what we are getting after two-roll mixing mill, will be undergo compression molding at high temperature and pressure. Then we are getting a sheet. The sheet is further going for the characterization techniques such as SCM, FTR, tensile, tear, aging, etc. Now coming to the resultant discussion, first one, the cure characteristics. So I developed two materials. First one, VR, vulcanized rubber. Second one, wool NR, wool natural rubber composites. And here the wool component I kept 50%. I mean 100%, 100% in the sense 50% rubber and 50% wool. I mix it together. So in the wool NR composite, it is a partial replacement of natural rubber with wool. And we compared the curing properties and we found this maximum torque is observed with the wool NR composites 35, which is it is around 4 times greater than vulcanized rubber. Minimum torque is also higher. T90, that is the curing time. Curing time, it is less for wool NR composites when compared to vulcanized rubber, because when we are adding some natural fiber to vulcanized rubber, the curing time is getting reduced. So this is the reason why T90, that is the cure time, is higher for VR and it is lower for wool NR composites. Coming to the mechanical properties, the tensile parameters such as tensile strength, strain, modulus, and tear strength were evaluated and compared. Vulcanized rubber is having higher tensile strength. However, when we are adding wool to the vulcanized rubber, the extensibility is getting drastically reduced. You can see in VR, it is coming around 1000%. Now, when we are adding 50% wool inside the composite, inside the wool, I mean inside the natural rubber, a drastic reduction, significant reduction is observed in the strain percentage, that the elongation is getting drastically reduced and it came down to 6.9. Modulus, the modulus of VR, it is very less, it's 2.3 because it is just opposite of strain. And when we are incorporated this wool NR, wool fiber in natural rubber, the modulus got enhanced. Similarly, I got almost double the tear strength when we added wool in the composites. Now, moisture content, we compared the data and hardness. So moisture content, when we added wool inside this one, hardness as well as moisture content is increased because wool is hydrophilic in nature and it absorbs moisture. So that's why we are getting higher moisture content. In case of hardness, when we are adding something to a vulcanized rubber, it increases the hardness. So natural fibers inside the natural rubber matrix generally increases the hardness. So this is the FTR spectrum of wool fiber, vulcanized rubber, unvulcanized rubber, and wool NR composites. Here I found there is no chemical reaction is taking place in between the wool fiber and rubber. However, it's a mechanical process of incorporating the fiber inside the matrix of natural rubber. Now, this is the scanning microscopic images, SCM images of the cross section of the composite. You can see the fiber pull out here and the fiber deformation is also, you can see very little number of voids are seen inside this. Most of the voids are closed and the matrix you can see here, it is natural rubber. The peculiar property of the wool fiber, that is the scales, that also visible in the SCM images. And when we compared the vulcanized rubber with the wool NR composite, the UV DR spectroscopy. So how much is the composite can absorb the UV radiation? That is a measurement of UV DR spectroscopy, and we found that when we are incorporating wool inside the vulcanized rubber, the vulcanized rubber is getting more capacity to absorb UV radiation. Because as we know, this natural rubber is having poor UV resistance. So it is evident that when we are incorporating wool inside the vulcanized rubber, the UV stability can be improved. This is a solvent diffusion data with water as well as toluene. Toluene is an organic solvent, water is a polar solvent. So while we compared this data, we found that toluene diffusion is higher for VR and is less for wool NR. Similarly, water diffusion is higher in wool NR composite in comparison VR. This is because VR is hydrophobic in nature and wool NR composite is hydrophilic in nature. So accordingly, we are getting difference in solvent diffusion. This is the aging data. Thermal and UV aging has been performed. After aging, we found that the aging causes, the thermal and UV aging causes decrease in the tensile strength, increase in the elongation, and decrease in the modulus. So when we are exposing this material, I mean composite material, to UV and thermal aging, considerable amount of change in physiomechanical properties were observed. And finally, we did biodegradation studies. So what we did, we collected the material, we buried in soil. There is a test known as accelerated soil biodegradation test. So we adopted that protocol, and after that we found that the weight loss is very less for VR, but for wool NR composite is higher. This is because the wool is biodegradable in nature. Now, in case of VR, why it is very less? Because when we are adding vulcanizing chemical, especially sulfur, it is forming cross-linking between the rubber molecules, that is isoprene units. Now, it's very difficult to break this particular bond, and the resulting material is having a very long polymer chain. So that's why bacteria and other microorganisms cannot break and degrade this material. So that's why the weight reduction in VR is very small in comparison with the wool NR composites. We developed some of the products. This is the highlight. So we developed some of the product. You can see the sheet what we have developed over here in the left side, the sandal, and you can see the purse, wallet, and bag, ladies bag, using this wool NR composite. Here the composition is, as I told you, it is 50% wool and 50% rubber. Now I made 2 mm thickness. So if you are, you can increase or decrease the thickness of the composite. For bag manufacturing, you need a better flexibility. For sandal, it may not be required. So the thing is that while we are changing the composition, instead of 50%, 50-50, you can take a 60-40 or 70-30. Accordingly, the flexibility of the composite will vary. Then it is possible to develop different type of products. So this is the thing. So coming to the conclusion, when we are adding wool fiber into natural rubber after vulcanization, we found higher torque but lesser cure time. Now, in comparison with the vulcanized rubber, wool NR composite possesses higher tear strength and modulus, water diffusion, moisture absorption, and UV absorption. The SEM images depicted dense pack of fiber inside the NR matrix, and FTIR analysis showed no specific chemical interaction. Now, coming to the last point, that the newly developed wool NR composite could find some potential applications in fashion accessories like bags, sandals, and other householdings. So, thank you very much. Thank you very much for giving me an opportunity for the presentation. And now the session is open for discussion.
Thank you so much, Dr. Dr. Seepa, for presenting and joining us today. Yes, and if any of the participants have any question, they can move on and ask the questions. Okay, Doctor, I don't think there are any questions, and if there are, we can discuss it at the end of the session. Thank you once again for joining us today.
Thank you very much.
Now I would like to call upon our next speaker, Mr. Shanti Babu Katta from Naipur, India, and he will be presenting his speech on synergistic in vitro antifungal efficacy of naringenin capped silver nanoparticles against Candida species. Hello, Mr. Shanti Babu, are you there?
Yes, hello, ma'am.
Yeah, most welcome.
So I'm sharing the screen. Ma'am, are my slides are visible?
Yeah, your slides are visible and you're audible as well. You can start the presentation.
So are my slides moving?
Yes, they are moving.
Okay. First, hello everyone. It is a great opportunity for me to share my research work on this platform. Thanks a lot to the Scholars Organization. Coming to my topic, synergistic in vitro antifungal efficacy of naringenin capped silver nanoparticles against Candida species. Of the presentation, now I will discuss inside of my research work, then formulation and its characterization, and in silico studies, and its in vitro antifungal activity, and finally conclusion. Coming to the inside of research work, according to CDC, the cutaneous candidiasis is the commonest skin infection. It affects all ages and genders. So of this infection, the Candida albicans is majorly responsible. It was estimated that 1% of all outpatients and 7% of all inpatients are affected by the cutaneous infection who visit to the dermatological clinics. So it can be primary cause for skin infection, or it can be secondary infection who are having the atopic dermatitis, psoriasis, and existing dermatitis. Here we can see the infection on foot and the hand. So with this, the silver nanoparticles were found to be potential antifungal solution. So coming to history of silver nanoparticles, it was used to treat potable water. It was used in wound repairing. It was also used as a counter irritant. And coming to its antimicrobial, antiparasital activity, it has broad spectrum of activity against various bacteria, fungi, viruses, and parasites. And in the literature, it was shown a synergistic activity when combined with the marketed amphotericin B, ketoconazole, fluconazole, and metronidazole. So these are a few products which contain silver products: Silvadine, Sildaflo, SSD, Thermogene, and nanocrystalline silver gel, etc. So it has also shown a great potential activity against Candida albicans and Candida glabrata. So with this, we wanted to explore the silver nanoparticles against the Candida albicans and Candida glabrata to combat the cutaneous candidiasis infection. So, but the challenges lies are when we synthesize the silver nanoparticles, the reducing agents are chemical reducing agents may lead to toxic byproducts. So further, the separation will be difficult. In this regard, we wanted to use some phytochemicals, but they are having a higher MS, well, less potent. So we by taking this, so we wanted to develop phytochemical with silver nanoparticles so that we can get the synergistic effect. So apart from this, we have also explored the in silico study. So because naringenin was not explored for the 14 alpha demethylase and ceramide, the 14 alpha demethylase is the popular antifungal target, and the ceramide is the ceramide present in skin barrier, skin lipid, which acts as a barrier for drug molecules and many other molecules. So this I will discuss in the further slides. So here I want to conclude that we wanted to synthesize silver nanoparticles by using naringenin, which is phytochemical, as the reducing agent. So both naringenin and silver nanoparticles can offer the synergistic effect. So with this, we have analyzed the naringenin by using RPHPLC method from the concentration range of 0.5 to 40 microgram per ml. So the column was used C18 column 250 mm, and we have also analyzed the silver content by using ICP-MS method. The ICP-MS grade silver standard was used and made calibration curve from the range of 62.5 to 100,000 nanogram per ml, which was further used in the formulation characterization for assay and release studies. Then we have synthesized silver nanoparticles by using naringenin as a capping or reducing agent. So we have obtained 32 microgram per ml of silver concentration and kept under heating at 100 degrees. When this solution was boiling, the naringenin, which was dissolved in 0.2 molar NaOH solution, added dropwise. When the color was changed from colorless to light yellow, the addition of naringenin solution was stopped, and the stirring was continued for more half an hour to cool it down. The color change indicated the formation of silver nanoparticles, and it was also characterized by using UV visible spectroscopy. The surface plasmon resonance peak was observed at 420 nanometers, which confirmed the formation of metal nanoparticles or silver nanoparticles when we have scanned between 300 to 600 nanometers. Then these nanoparticles were also characterized for particle size and jitter potential. The particle size was found to be 32 nanometer, and the PDA was found to be 0.2. Coming to the jitter potential distribution, it was found minus 32 millivolt. So according to DLVO theory, the colloidal particles, which shows plus or minus 30 millivolt, we can expect the stability of the nanoparticles. Then coming to, we have analyzed these silver nanoparticles by using a transmission electron microscope. So we have found the particles are in spherical shape and with no aggregation. So then the silver nanoparticles were also analyzed for assay, in which the naringenin content was found to be 100%, and the assay of silver ions was estimated to be 96%. So then these silver nanoparticle solution was converted into the topical dosage form by using carbopol ultrase, which was 0.9%, and the pH was adjusted to 7 by using triethanolamine. So this topical dosage form further characterized for rheology and texture analysis, and it also compared with the marketed methanazole nitrate gel. So how viscosity and its texture. So we can see the topical dosage form of silver nanoparticles has shown decreasing viscosity with growing shear rate, representing the shear thinning system. And coming to the texture, it was estimated to be 200 grams. The lower the firmness represent the easier spreadability upon topical application. So it was also comparable with the marketed formulation. We can see these red and blue lines. Then it was the topical dosage form was also characterized for pH, and pH was found to be 7, that is acceptable for skincare products, and the gel product was estimated for naringenin content, which was found to be 105%, and the silver content was found to be 95%. For the most of the drug products, according to ICH and USFDA guidelines, the drug product should be in the percentage range of 90 to 1%. So our product was in the within the range limits. Then after this, we have also explored in silico study of naringenin and naringenin silver complex, and with the methanazole nitrate. So we can see the lanosterol 14 demethylase protein and with the different naringenin and the naringenin silver complex and the meconazole nitrate. In the next slide, we can see its docking scores and the hydrophobic interactions. So when we compare the naringenin silver complex docking score with the meconazole, so it was almost similar, and the both of them have shown some hydrophobic interaction. So which concluded that, which predicted that the naringenin silver complex can have the potential affinity towards the 14 alpha dimethyl, which further shows antifungal activity. Next, the naringenin complex was also explored with the ceramide, as I have discussed in the initial slides. The ceramide present in the skin barriers, which acts as a barrier. So we want to see how much affinity is there with the naringenin and naringenin complex towards the ceramide. So if the naringenin complex having more affinity towards the ceramide lipid, so we can predict that these naringenin silver complex may show higher permeation efficiency than compared to the pure naringenin. So we can see here docking scores. Here naringenin was showing -3, and the naringenin silver complex was showing -2. So which is more than the naringenin. So with this, it was also confirmed that silver complexation of naringenin has induced conformational alterations in the docking study, and it also showed hydrogen bond along with the pi-pi stocking interaction, which was not present in the pure naringenin. So this has predicted the more permeation of the skin naringenin silver complex than the pure naringenin. So with this, we have conducted ex vivo permeation studies by using the mice skin with the help of Franz diffusion cell. So the pure naringenin gel has shown that 18% of permeation, which was significantly less than the naringenin silver nanoparticles topical dosage form, which was found to be 36%. With these values, we have also calculated its flux value. Flux value will be demonstrated in the terms of microgram per centimeter square per hour. So the flux value also higher than the this pure naringenin topical dosage form. So on the other side, we have also estimated skin retention efficiency of our formulation and compared with the simple naringenin gel. So obviously the naringenin silver nanoparticles loaded topical dosage form has shown 37% retention in the skin, which was required for localized treatment, and the pure naringenin topical dosage form has shown only 8% of skin retention, which is 6.4-fold lesser than the silver nanoparticles, naringenin silver nanoparticles topical dosage form. So finally, we have conducted in vitro antifungal studies. Here we can see the this naringenin silver nanoparticle topical dosage form was having lower MIC values when compared to pure naringenin and trisodium citrate silver nanoparticles topical dosage form. So with these values, we have also calculated FICA index, which is used for calculating synergies, whether the product is giving synergistic or additive or antagonistic effect. So we have found that the values below the 0.5, so it can be concluded that the product has shown the synergistic effect. So coming to the conclusions, the silver nanoparticles were synthesized successfully by using phytochemical, and they were loaded into the topical dosage form, converted into topical dosage form, and the in silico study has revealed the potential affinity of naringenin towards the naringenin silver nanoparticles complex toward the 14 alpha dimethylase, the popular antifungal target. Then ex vivo permeation study revealed that these naringenin silver nanoparticles has shown increased permeation and retention in the skin, which is required for localized action. And the naringenin loaded topical dosage form has qualified all the desirable pharmaceutical testing parameters which were recommended for topical dermal products. And the in vitro fungal activity has shown the synergistic activity of naringenin silver nanoparticles when compared to pure naringenin. So all these results has gave us the confidence to do the further in-depth in vivo study under a set of stringent parameters. So for translating this product into a clinical viable antifungal product. My acknowledgement, sincere thanks to the Professor Shailendra Saraf, Director, NABER Hyderabad, my guide, Dr. Jitendra Madan, and my seniors, colleagues, and my juniors. Thanks a lot, everyone. Okay. Hello.
Yeah, organizer. Julelos. Pose. Pose. Anyone there?
Hello. Hello, everyone. Welcome back after the reflection break. I would like to call upon the next speaker. Hello, am I audible? Can you please, one of you, respond to me? Okay, thank you. Now I would like to call upon our next speaker, Mr. Hafiz Muhammad Tawfiq from Quaid-e-Azam University, Pakistan. And he'll be presenting his topic is reduced graphene oxide zinc sulfide nanocomposite decorated with silver nanoparticles for wastewater treatment by adsorption, photocatalysis, and antimicrobial action. Hello, Mr. Hafiz, are you there? Can you hear me? Yeah, hello, Muhammad Tawfiq. As you there, will you be able to present your speech now? Please respond to me. Hello, Mohammed Tawfiq. I would like to request you to please click on join audio so that we can also hear your voice. You are not audible to us. Please click on join audio. Once then only your audio can be connected. Yes, your slide is visible now, but you're not audible to us. Hello, Mr. Tawfiq. We can see your slides, but we can't hear you. Can you please connect the audio? Hello, Mr. Tawfiq. Is there a network issue as we are not able to connect with you and we can't hear you as well as see your slides? Please let me know in chat if you are facing an issue. I will help you accordingly. I don't think then we can connect to Mr. Tawfiq. I think he's facing some network issues. So now I'd like to move on with our next presentation. Mr. Tawfiq, if you can hear me, please respond to me. Okay, in that case, I would like to move forward with our next presentation. Now I would like to call upon Professor Atefe Ruzi Taleb from Amir Kabir University, Iran, and she'll be presenting her speech on performance of green membranes in harsh conditions. Hi, hi, Professor. Are you ready for your presentation? Can you hear me?
Yes, hi.
Most welcome.
Can you hear me?
Yeah, we can hear you.
Okay. I can share my PowerPoints.
Beside check, there is share screen there. Beside check, then share screen. Press the share screen, then you can share.
Okay, I can share my PowerPoint. I don't know what.
Okay, first.
Okay.
Yeah.
You can you see?
We can see. You can switch your screen to your presentation slides. Now the
Yes.
screen I can see is of the Zoom meeting actually.
Okay. Hi. Hi everyone, and good afternoon everyone. My name is Ata Feruzi Talab, and I'm a PhD student in chemical engineering at Amerikabi University of Iran. So my thesis about the performance, my thesis, PhD thesis about the preparation tubular ceramic membranes. So as you can see, my presentation is about the performance of green membranes in harsh conditions. And my supervisors is Professor Ali Kargeri and Mansour Soleimani in Amerikabi University. So industry and academic are both under pressure to create more sustainable alternative to several conventional processes. But to the growing awareness of global issues like water and fossil fuel concentration, so the green technology is investigated. The green technology refers to a technological concept that aims to elevate or rectify the consequence of human behavior on the natural environment.
Excuse
So,
me, sorry to interrupt. Actually, we can't see your slides. Can you please move your screen? And can you open your screen where you have the slides? Actually, the screen we can see is of Zoom meeting actually.
can you see? Can't you see my presentation?
No.
No.
What?
Okay. Let me help you. You can first open open your PPT, and then you can start reading it out from there. And actually, your screen is off now. Zoom meeting.
Okay.
Open your current screen.
Are you? Can you see?
No.
No.
Please let me stop sharing and.
Once click on stop sharing.
Okay.
Now, can you please open your PowerPoint? Can you find your PowerPoint and just let it be in your recent tabs? Just open your presentation, then you can come back to this original Zoom meeting, and then if you will click on share screen, it will give you option of which screen you can share. At that time, you can just click on
Okay.
yeah.
I use the websites of the Zoom, not software.
Okay, no problem. In that case also, just
Okay. Can you see?
yes.
Yes.
Okay. My presentation is about the performance of green membranes in harsh conditions, and my supervisor is Professor Ali Kargeris and Mansour Soleimani. My PhD thesis is about the preparation of the ceramic membranes in tubular to use in a DCMD process, means direct contact membrane distillation. And now my presentation about performance of green membranes in harsh conditions. Industry and academics are both under pressure to create more sustainable energy instead of the conventional energy, because global issue like water and the lack of renewable energy the green technology is investigated. So the green technology refers to a technological concept that aims to all elevate or resistivity the consequence of human behavior on the natural environment. And this technology encompasses a comprehensive range of advancement in terms of techniques and material, from energy generation method to non-hazardous cleaning agent and material. And the aims, the paving the way for a sustainable society for future generations. But the membrane technology generally is an environmental friendly and sustainable process. But recently a study, and you know that the membrane fabrication process is far from green, because annually this process is produced more than 50 billion liters of wastewater that is contained with a waste solution, polymer and membranes, and the hazardous solvents or non-solvents. And these toxic solvents and non-solvents are wasted and enter to the environment. So membrane preparation process produce 50% of the overall waste. And so choosing the material in this step is crucial. Replacing non or low toxicity solvent with a common solvent, and replacing biopolymer or a green polymer instead of the fossil polymer, and choosing the inorganic membrane can help the membrane process closer to environmental friendly. So Paul Anastas and John Warner published the green chemistry laws of the green chemistry set as of the 12th law. And this law pertains to novel chemical synthesis and procedures. And you know that the membrane classified into the two categories, polymeric membranes and inorganic membranes. And the more this membrane pass the number of this law, this closer they can be to the friendly environmental. So in this presentation, focusing on this rule, we review recent research to obtain an overview of green membranes and their use in a harsh condition. The polymeric membrane, same as other material in the world, have advantages and disadvantages. Simplicity, low energy, flexibility, and ease of scale up and control are advantages of the polymeric membrane, but the preparation of polymeric membrane is not environmental, as we mentioned, because using the non-decomposable or lime-decomposable polymers, toxic solvents and the toxic non-solvents are using in this process. And the recently research non-solvent is utilized in membrane manufacturing roughly 10 times greater than a solvent. So in a greater detrimental impact on the environment. Choosing the biopolymers and the green polymers is the best choice for the help the membrane processor can closer to environmental friendly. Biopolymers such as cellulose acetate, chitosan, polylactic, alginic acid, polyhydroxyalkanate, polyethylene glycol, and polycaprolactam. And the green polymer used in some applications such as metal removal, dye removal, water purification, biomedical processes produce renewable energy, and the gas separation. So the another solution is using the biosolvent. The common solvent is used in the membrane process is DMF, NMP, THF, and chloroform. These solvents are very toxic and harmful for a human and environmental. So choosing the biosolvent is another choice, but it's challenging for the scientists because important properties of the biosolvents and the common solvents is very different. For example, solubility, viscosity, polarity, and boiling point and dielectric of these solvents are very different, and in fact, effects on the membrane structure, and finally effects on the membrane performance of the membrane separation processes. For example, common biosolvents in membranes, methyl lactate and ethyl lactate, are the biosolvents in membranes. and it's used for a cellulose acetate polymer. And the common solvent, such as DMF, DMA, acetone, and dioxane, is a common solvent for a cellulose acetate polymer. And you can see in the table the Hansen solubility of parameters of these solvents are very different. So the membrane structure is different. You can see in the picture, you see the SCM image of surface and cross section of the cellulose acetate membrane with a solvent and methyl lactic solvent and the DMF solvent. You see that the membrane structure is very different, and these effects on the membrane performance of in a membrane processes and in the membrane structure. So another topics and the main topics in presentation is about the performance of green membrane in harsh conditions. You know that the membrane having some fields and the harsh conditions such as high temperature and high pressure is the harsh conditions, and these figures is obtained by the Scopus database and thereon by the VOS viewer software. And you see that the high temperature and the high pressure and the high total in a harsh condition is very less than the other membrane fields, such as separation and application. And you know that the polymeric membranes are not have a good performance in a membrane processes in a harsh condition. So the another choice is the inorganic membrane. The inorganic membrane based on the metal and oxide and the carbon, and the carbon-based steel membranes and the ceramic membranes is the kind of this inorganic membranes. Inorganic membranes, same as the polymeric membranes and the other metal in the world, have advantages and disadvantages. High mechanical temperature and chemical stability is the advantage, are the advantage of the inorganic membranes, and the brittleness and the high capital installation and modification cost is are the disadvantage of the inorganic membrane. So it's important to us that at least 10 of the 12 principles of green chemistry apply to inorganic membranes. So these membranes can help the membrane processes can closer to friendly environmental. But we need a range of parameters to check a harsh condition. The limitation of the polymeric membranes in the harsh condition is the good criteria for our to determine and investigate the harsh condition. And this table that can you see is published by our team by overviewing the 100 of the over the 100 of the recent research, and some parameters such as plasticization, the polymeric membrane, and the TG of the polymeric membrane is the essential parameters to gas separation and wastewater treatment by membrane. Also, swelling and membrane falling effects and membrane performance after washing by an acidic and alkaline solution is very important for us. So this range of the parameter is obtained by our team. And to following slides, I focus on the ceramic membranes, because by overviewing of this recent research, we found that carbon-based membrane and steel membranes don't have a good performance in a harsh condition, and the ceramic membranes are good performance in a harsh condition, and we focus on the ceramic membranes. So high pressure is one of the harsh condition, and the high mechanical of inorganic membranes is essential for us because the high mechanical strength of ceramic membranes is attributed to the high centering temperature, and the raw material additive and the preparation method of the inorganic membranes effects on the mechanical strength. For example, by adding the feldspar as an additive to a solution, the mechanical strength of the inorganic membrane and the ceramic membrane is increased. and is by using some preparation methods such as a piercing method, the mechanical strength of is increased. The another parameters is high temperature. Some membrane processes needs a high temperature. For example, membrane reactor, separation of lignin from kraft, cooking liquor, or and the gas separation. You see that in the right image. You see the extractor and distributive concept of the membrane reactor. The membrane reactor needs a high temperature, and this process is done over the 600 Celsius, and the separation of lignin from kraft cooking is done over the 100 Celsius. And in the gas separation, recently research shows that by increasing the temperature, the permeability of the some gases such as helium and H2 is increased a slight, but by increasing the temperature, the permeation of the some gases such as CO2 increasing sharply. So selectivity of these gases are high, and it's important for a membrane separation and for a gas separation by membranes. So another parameter is pH of the environment, and some processes such as NF, UF, and MF means nanofiltration, ultrafiltration, and microfiltration influenced by the pH of the environment, and the pH effects on the membrane fouling, and the membrane fouling is produced by the filter cake, and the membrane surface properties and the feed composition effects on the filter cake. And the another parameters is the pH isoelectric point, because the recently research shows that if the solution and the process are done in the around the pH isoelectric point, the average specific filtration resistance and the cake porosity is maximum and minimum. So the effect of pH parameter in the formation of filter cake and the permeate flux in the membrane processes is very noticeable. So finally, we conclude. For conclusion, first, due to the solvents and polymer solution waste, the manufacture of the polymer membrane increase environmental contamination. And second, due to their raw material and solvents, inorganic membrane are therefore environmental friendly than a polymeric membrane. And polymeric membrane don't have a good operation in a harsh condition, such as a high temperature, high pressure, or acidic and alkalic environment. And finally, among inorganic membranes, ceramic membranes perform best in a harsh condition. So recent studies additionally demonstrate that the membrane industry and the science are working together to increase ceramic membrane studies in order to address the environmental issue. And finally, thanks for your time and attention. And anyone have a question, I'm happy to answer that. Hi.
Thank you, Professor Atefe, for your presentation.
Thank
If
you.
any of the participants have any questions, they can go on and ask. Okay, I don't think there are any questions. And if there are, we can discuss it at the end of the session. Thank you once again, Professor.
Thank you.
Now I would like to call upon Mr. Hafiz. Can you hear me? Can you join us now? Please try connecting the voice and the visuals as well. Can you please try and let me know that you'll be able to present now?
Am I audible to you?
Yes, you're audible.
Okay, let me share the screen.
Okay. Yes, we can see your screen as well.
Okay, let me zoom the screen. Am I audible to you people?
Yeah, it is using and you're audible as well. Most welcome, Mr. Hafiz. You can start your presentation. Mr. Hafiz, we can't hear you actually. Can you please connect your audio?
Right now. Hello. I think there is some problem in the connection, so hello.
Yeah, we're hearing you.
I think the voice is one second.
Actually, your voice is echoing so much. Can you please change your place or can you sit at somewhere else?
Exactly, change.
Can you continue now or should we move on to the next presentation? Please let me know as we have to be on schedule.
I think there is some problem in the voice.
Yeah.
Am I audible right now?
Hello.
Am I audible?
Yeah, you are audible.
Okay, this is the story of graphene when I selected graphene for the two layer of hexagonal structure of sp2 bonded carbon atom.
Okay. Can you please use headphones? Maybe that will improve the quality of the voice. Actually, your voice is not properly audible to us. Can you please use a headphone or?
There is a problem, so I want to mute this.
Okay, sure. You can. Okay, you can continue.
voice is echoing.
Mr. Hafiz, we can't hear you actually. Nothing is audible to us.
I think there is a problem, so I want to skip this.
Okay, sure, you can join after some time and then we can try again. But for the time being, I would like to proceed with our next presentation. Thank you for understanding. Okay, I would like to present, I would like to introduce our next speaker. We would like to call upon Professor Noor Fatima Yahya from University Sains Malaysia, and she'll be presenting on the title of Rapid Ultrasound Assisted Emulsification of Microsolid Phase Extraction based on molecularly imprinted polymers for HPLC DAT determination of bisphenol A in aqueous matrices. Hello, Noor. Can you hear?
Hi. Can you hear me and can you see my slide?
Yes,
Yeah, we can hear you and we can see your slides as well.
All right. So may I start now?
Yeah, you can start.
All right. Thank you very much, Ms. Chairperson. So, Assalamualaikum warahmatullahi wabarakatuh and good day, everyone. I am Dr. Noor Fatima Yahya from Advanced Medical and Dental Institute in University Sains Malaysia. So my expertise is in separation science or in analytical chemistry. So therefore, I use this polymer for the separation technique, or which we can call as micro extraction technique. So the title for my presentation today is Rapid Ultrasound Assisted Emulsification Micro Solid Phase Extraction based on molecularly imprinted polymer for HPLC determination of bisphenols A in aqueous matrices. So I believe some of you might came across this solid phase extraction. So this is the new generation of solid phase extraction, which we call as micro solid phase extraction. All right, so I would like to give a brief introduction on the bisphenol A. I think everyone already know about the harmful or the adverse effects of bisphenol A. It's known as endocrine disruptors due to its ability to exhibit estrogenic activities in human body, and the European Union has set some limits of this BPA in food samples and environmental samples. And nowadays, research are more focusing towards these bisphenol A analogs like bisphenol B, bisphenol F, bisphenol E, because the manufacturer, they actually replace this bisphenol A, because bisphenol A, we already established the guideline for this bisphenol A. But for other bisphenol, there are no establishment of the guidelines or toxicity study or thorough toxicity study about other bisphenol analogs. So that is why now I think it's time for the researcher to moving towards analysis of other bisphenol A analogs. So the effect of, I would like to brief a little bit on the effects of bisphenol A. It can enhance the risk of type 2 diabetes mellitus, hypertension, and dyslipidemia due to its ability to disrupt pancreatic beta cell function. And it has the estrogen-like activity, and its blood levels in men and women are associated with reproduction dysfunction, endometrial hyperplasia, recurrent miscarriage, abnormal karyotypes, and polycystic ovarian syndrome. Okay, so there are a lot of adverse effects of this BPA. There have been reported a lot of literature can be found on this, the adverse effect and harmful of this BPA compound. So that is why this is one of my master student by research project. So this research was conducted in 2017. So, but I believe this research is still relevant to be presented today. So okay, this is the analytical process, usually in how we conduct the chemical analysis. When we have any sample to be analyzed, this is the process that will be conducted in the laboratory. First, we will do the sampling, and then sample preparation, and then the separation, detection, and data analysis. So why the researchers are focusing towards the sampling and sample preparation is because they covered more than 80% of analysis time. So the researchers are now moving towards how to produce a rapid analysis or rapid sample preparation so that we can cut time, save time. Of course, when we save time, we save the cost, and also towards the more environmentally friendly system for sample preparation. So other than that, the sample preparation, some of the conventional sample preparation, they consume a large amount of organic solvent or toxic organic solvent. So this is also the interesting scope of research that we can actually explore to replace the hazardous or toxic solvent with more greener solvent like ionic liquid and also deep eutectic solvent. This is one of the current research that I have been, or we, and me and my research team actually explored into these things. So therefore, it is very important to find a suitable sample preparation method as great influences toward the reliability and accuracy of the analysis. So for anyone who did not very familiar to the analytical chemistry or chemical analysis with the instrumentation, this is how we conduct, how we analyze the sample, how we do identification and quantification of this compound in the HPLC or chromatographic system. So in the perspective of sample preparation is to prepare the sample to make it amenable for separation or detection part of the analysis. So you just imagine, for example, you want to analyze this bisphenol compound from river water or from food sample, canned food sample. So you need to prepare this sample so that the sample is ready to be analyzed by the instrumentation, and you can get the limit of detection or limit of quantification that you want to see. Okay, so you have to remember most of these contaminants, they actually present at very low and minute concentration level at that sample. So you need a very sensitive methodology in order to detect all those compounds. So the key function of sample preparation, okay, the micro extraction, the sample preparation, or some of the, in some of the journal, they might put sample pre-treatment. So they are the same. We prepare the sample before we introduce to the instrumentation. So the key function is to convert the analyte. So in this case, is the bisphenol from the analyte inside the sample, okay, from a non-compatible medium, which is the sample, into an instrumental compatible with analytical methods. For example, you have HPLC, you cannot directly inject all those samples to the HPLC because you will block the column, you will damage the column. So you need to prepare and make sure that there are no bigger particles or interferences that will interrupt with your detection. And other than that is to remove the major interferences, of course, because we are doing the quantitative analysis of the trace element. So you are dealing with part per billion level. So we need to remove major interferences so that you can have very good peak identification. You can integrate a good peak area from your chromatogram, and you can give an accurate data. Okay, and of course the sample preparation, when you have eliminated the interferences, you do a very good sample pretreatment. Of course, you can consequently improve the sensitivity and limit of detection. And other than that, this sample preparation, the ultimate goal is to isolate the bisphenol, to extract the bisphenol, and to pre-concentrate the bisphenol so that you can get a very good sensitivity of the detected compound. So when you design a sample preparation, you have to understand the properties of the analyte. What are the pKa? What are the polarity, the solubility of the analyte so that you can design your sample preparation? How you can design your sample preparation, or you can design your adsorbent. For the solid phase extraction, we are using the adsorbent to extract the analyte. So you can design the analyte according to what sample state. Do you have a liquid sample, a solid sample, and the sample matrix, whether it is complicated sample like wastewater or very simple matrix like drinking water. But when you deal with drinking water, the challenge is to get a very low detection limit, because in drinking water, the limit for the toxic compound is lower than that in the environmental sample or wastewater sample, for example. And the representative sample, for example, if you're looking to collect the sample, river water sample, how do you know that which location should you do your collection? Okay, so this should be representative of that river water. So you have to plan very well, and of course the available instrument, what you have, whether you have the GC or LCMS or LC-UV system, and the concentration level. This is very important. So when you design a method or to do chemical analysis, you have to know what are the maximum residue limits. So if you do a method, if you develop a new method and you cannot detect at this maximum residue limit, there will be no use, right? There will be no, the impact will be lower, because we want to detect so that we know that whether it has exceed the safety level or not. Okay, and then for the detection. Okay, so the conventional sample preparation that I mentioned before, this is one of the example, is the liquid extraction. So this liquid extraction is very easy to be operated, and I believe some of you might have used this separatory funnel, where you have to mix the two immiscible solvent, one immiscible solvent with your sample, aqueous sample. So you will shake, and you will release the gases. So this is the liquid sample, liquid extraction. So based on this technique, the liquid extraction, okay, so you will need to use the immiscible solvent, right? So most of the time, the one or the laboratory, the chemists, they will use like dichloromethane or hexane. So this is a very toxic, and you will use a relatively large amount of this solvent. So you can see how big the separatory funnel. Okay, so you will need a lot of solvent. So the disadvantages of this technique LLE is laborious, because you need to shake every time, and sometimes you need to shake multiple times, and it consumes large amount of organic solvent, relatively time consuming, and sometimes when you shake, it will form, the emulsion will be formed, and this will actually result in poor recovery of your analyte. Okay, and for this configuration, it is very difficult to be automated. And the researchers have seen the drawbacks in the LLE. So what the researchers did was they developed a new technique that we call as solid phase extraction. So you can see from this configuration, it become more miniaturized, and it has like the adsorbent. Okay, this is what the white color is, the adsorbent. It's being packed inside the solid phase extraction cartridge. So this is what we call as cartridge SPE. Okay, so you will need to condition the cartridge first, okay, to activate the adsorbent and to remove all the impurities. Okay, and then you will load your sample here. So this adsorbent is actually the functional group or the selective adsorbent that will capture your targeted analyte. Okay, so by having a certain steps in the SPE, okay, you can have a very good recovery. So the advantage is includes a high selectivity, high recovery, high reproducibility, and capability to extract variety of matrices. Why? Because they are the commercially available adsorbent or the cartridge, different types of cartridges are available, okay, from the manufacturer. So you can choose, depend on your analyte, whether it is a polar analyte, non-polar, mixed ion exchange analyte. So you can just purchase them and do the solid phase extraction. However, there are some drawbacks of the solid phase extraction. Okay, so these cartridge, they are meant to be used once only. It cannot be reused. Okay, and because of that, it contributed to the relatively high cost analysis if you have many sample, or if you want to do like routine analysis. Okay, so you have hundreds of sample, then it could contribute to very high cost. And like what I've mentioned before, you will have several steps to do this SPE. So sometimes it will contribute to the time-consuming analysis, okay, and involve multi-steps of operation. So the objective of this study is to develop and validate this ultrasound MIP micro SPE method, which we call as micro scale extraction or micro extraction technique. And this technique, okay, or this method has been applied for the determination of bisphenol A in water, beverages, and aqueous canned food. So a little bit introduction on the MIP. So the compound that has been processed through the process of imprinting, where it allowed the synthesis of artificial receptor of the target molecule. Okay, in a simple words, that this material of MIP or molecularly imprinted polymer is a very selective material because you are using the bisphenol A, okay, as the template. So it will only recognize a structure that's similar to the bisphenol A, okay, or other analogs or similar structure can only be captured by this material. So that is why it will give you highly recognition sites, and it is very selective towards the compound that you use as the template monomer. Okay, so for example, here you put the template monomer, so only the template can be captured by this polymer. That is why we call it as molecularly imprinted polymer. You imprint the template molecule or the targeted analyte that you want to extract. So this is what we call as the micro solid phase extraction. If you still can remember, the LLE is inside the separatory funnel, and the SPE, you are using the SPE cartridge. Now we are moving towards micro SPE or miniaturized solid phase extraction system. So you can see in this figure, all right, there is a triangle shape of membrane. So actually we have to seal this membrane. Okay, we have to use the heat sealer, this sealer. Okay, and we seal. So inside this membrane, we put the molecularly imprinted adsorbent. So if you can see, this is only a schematic diagram, but in the real photograph, this is actually very small. So we need to use forceps to actually seal, okay, the edges of this membrane, and we put only few milligrams of molecularly imprinted polymer inside this. Okay. So how you are going to do extraction is just that you just put this one, okay, this membrane enclosed with this MIP, you put inside your sample, and then you on the magnetic stirrer, and after some time, after complete the extraction time, you will take out this membrane and put inside the desorption solvent for the enrichment, where you dissolve this BPA, okay, in a very small amount of organic solvent. Okay, and then that organic solvent will be directly injected to the HPLC. So the tips and tricks, because I am going to share with you my experience, because I completed my PhD in 2014, and until now I am working on development of this micro extraction. So I think this is a very useful tips if you want to try this micro SPE. So first is you have to consider the hydrophobicity of the membrane. So the membrane is hydrophobicity because it made from PTFE. Okay, that membrane made from PTFE. So sometimes when you stir inside the sample solution, it can go up and it can go around. Okay, it's not consistent. Okay, it will produce an inconsistent result. Okay, so And then some of the researcher, I think I just read one of the publication that they put something to weight the adsorbent so that the adsorbent will become heavy and it will become static in the sample solution. This is one way on how you can overcome this hydrophobicity of the membrane issue. And then you need to condition the membrane similar to the SPE, where you need to condition first, okay, the adsorbent, and then you need to optimize the extraction time. So the advantage, so I think this micro SPE is not new. It has been developed, I think, long time ago, maybe 2012, if I'm not mistaken, by Professor Bashir. Okay, so the superior advantages of this micro SPE is very simple operation. The membrane-based micro SPE device also function as filter. So sometimes, if you look at the configuration, so if this is the membrane similar size to the the membrane filter that you use to filter your mobile face, the round shape. Okay, so it is actually can act as filter to remove major interferences, and you will need only little amount of solvent and adsorbent consumption, and you can reduce the metric effects. Okay, however, you can reuse this adsorbent or this micro SPE device. This is what we call as micro SPE device. It can be reused, but you have to make sure that you do some washing. Okay, but the possibility of carryover effect is always there. Okay, you have to consider about the carryover effect, and because of the membrane, the sample will pass through the membrane before it interact with the adsorbent inside this membrane. Okay, so they will take a longer time to achieve the equilibrium. Okay, and also to complete the mass transfer. Okay, of the bisphenol A or your target analyte from the sample solution to adsorbent. So this is how we prepare the membrane. Okay, so we cut into 1 centimeter, so it is very small, 1 centimeter, and then you fold like this. So the reason why we do this triangle shape, because for triangle shape, we only need to seal at 2 edges here. All right. So this is the procedure. It's a very simple procedure. Okay, when you have your sample here, okay, I use 1 octanol. Okay, 1 octanol is actually very significantly improved the recovery, because we understand or we observe that when we put 1 octanol, it's actually enhance the extraction by producing the micro droplet, okay, that carry the analyte inside this membrane. Okay, and then the sample was sonicated, and then we put the micro SPE device here, and then sample was vortex for 4 minutes for the extraction, a rapid extraction, so that the analyte can be transferred into the adsorbent. And then when we have completed the extraction time, we will take out the adsorbent or the micro SPE device and put inside the desorption solvent, which we use ethanol in this case, and then sonicate only for 1 minute, and you can transfer to HPLC for analysis. So of course, when we develop a new method, all the parameters that we use inside this micro extraction, we need to optimize. So at this time, we are using the one variable at one time approach. Okay, so this is all the parameter. So before that, at this moment, okay, for master level students, this molecularly imprinted polymer is actually we actually purchase from the company. If I'm not mistaken, a Superco company. So we purchase this polymer, molecularly imprinted polymer. We purchase in bulk, and we use 4 milligram for each of these micro SPE device. Okay. So these are all the parameters that need to be optimized. So you can see only 4 milligram of MIP was required for each analysis. So the normal weight or the amount of adsorbent inside the SPE usually like 100 milligram to 300 milligram, depends on the capacity of the cartridge. So you can see the micro scale extraction only require 4 milligram of adsorbent. Okay, and the desorption solvent ethanol, volume of desorption solvent only 200 microliter, so that we can give enrichment to this bisphenol A prior to the analysis. And we also optimize the ionic strength. So the ionic strength is very important, especially towards the polar molecule, because when we add the salts, we actually reduce the solubility of polar molecule, and it will be easier to extract this polar molecule, okay, when it's reduced its solubility. And we also optimize the extraction time and of course pH, okay, and then the desorption time and also sample volume. So this is the analytical performance, and we can use this micro SPE device up to 11 cycle, meaning that after 11 cycle, the recovery is less than 80%. So this is the linear range. If you see here, okay, this is microgram per liter, which is the part per billion level. So from 0.5 to 700 part per billion is the linearity range. And you can see the LOD can goes down to 0.07 part per billion level, which is, I think, very good for HPLC UV, a conventional HPLC. So we also detect these bisphenols in the river water, okay, and we do the recovery study. Recovery is to actually check the accuracy of the method. So all produce a very good recovery, more than 80%. And we also compare to the other method as well, all right. So you can see here, this is the example. First is this is the typical chromatograms of the BPA. Okay, for the first is the water sample before pretreatment. This is the without any pretreatment. But when we run with the micro SPE, ultrasound micro SPE with MIP, so this is the peak that we have. So you can see the scale still maintained, but the peak is actually increased significantly. All right, so this is how to explain the functionalities of this sample preparation or the micro extraction that we use here. And C is the real aqueous liquid in the canned food sample. We also detect the BPA at 14.59 part per billion level. So this one we compared with other methods. So you can see the LODs is quite good compared to other methods, okay? So for the conclusion, the proposed method has been successfully developed and applied to several types of aqueous matrix. The proposed method gives good selectivity towards the target analyte. And for future direction is maybe towards the automated of micro SPE system. That is one thing. Number two is what my research team is now working on is to synthesize the molecularly imprinted polymer using the deep eutectic solvent, okay, as the monomer. All right, that is one of the research scope that we are going carry forward, okay, in our future direction. So I would like to acknowledge the Ministry of Higher Education Malaysia and my institute, Advanced Medical and Dental Institute in University Science Malaysia. So this work has actually published in Talanta, Q1 paper. So I think this is also one of the good paper in the separation sign or in analytical chemistry. So I think, I'm also open for any collaboration. So I can be contacted through my email, norfatima@usm.my. So I think with that, thank you very much. If you have any question.
Thank you, Ms. Noor Fatma, for your presentation. I hope all the participants have gained critical insights from that, and they can ask questions if they have any. In case there are no questions, we would like to move to our next presentation.
Thank you very much.
Okay, now I'd like to call upon our keynote speaker for our next presentation, Dr. Sabrina from East Paris University, France, and she'll be presenting on the title Development of Tailor-Made Drug Delivery System Based on Polyelectrolyte Multilayer Capsules. Hello, Dr. Sabrina, can you hear me?
Hello everyone. So let me just share my screen.
Yeah, okay.
It's okay for you to see it?
Yeah, we can see your screen.
Great. So hello everyone, hope you are doing well. So I'm Sabrina Belbekouche. I am an associate professor at the East Paris Institute. Oh, and I work at the, it's okay. East Paris Institute of Chemistry and Material Science. So let me show you here our nice institute, as you can see here, located in Paris, in which we are working in different unity of expertise. So some of them are listed here, as you can see. Sorry, just 1 minute. No, sorry, I just want. Okay, so I'm working in the last one, so which concerns the chemistry for the healthcare. So indeed, I am a physical chemist, and I'm interested in developing new innovative drug delivery system. And for this, it's very important to understand problem which can both come from both physical chemical point of view and biological point of view. So for the first case, the more general problem that can be seen is related to the poor stability of the drug, or simply due to the poor solubility of the drug in the aqueous media. Concerning the biological part, the major problem is related to phagocytosis phenomena, or simply due to no or low incorporation of the drug. So developing drug delivery system is known to allow overcoming such issue. More precisely, it's very important to develop drug delivery system with well-controlled property. For instance, controlling the size and the surface characteristic of the particle has been proven to allow modulating the biodistribution parameter. So in this context, I am interested in developing a strategy which allow controlling several parameters all at once, namely to control the size, the chemical composition, the surface charge, and the aggregation state. For this, I use a strategy combining both the templating method and the layer-by-layer approach. So as template, I use particle, which could be either porous or non-porous. And the interest of using particle as template is to be able to control the resulting size of the particle and the polydispersity of the particle. And the interest of using the layer-by-layer technique is that technique allows controlling the chemical composition of the particle, and also the mechanical property can be controlled by removing the template. So we can consider the possibility to prepare polyelectrolyte capsule or simply polymer capsule. And this is very important to control the mechanical property of the particle, because it has been shown in the literature that it can favor the crossing of the particle in some biological barrier. The main interest of this strategy is that there is no specific equipment which are required, no organic solvent are required, and we can easily functionize the layer. So now I would like to show you some specific application that we have. So I will show you the interest of the system that we are developing to use them as anti-cancer or antibacterial, or to try to find or to try to propose a solution to treat a specific kidney disease. Okay, so let me show you the first case. For this, we collaborate with the team of Professor Gascon. So Professor Gascon is a biologist. She's interested in finding a solution to treat the pancreatic cancer. So in few words, the pancreatic cancer has very poor prognostic. And the main problem of this cancer is, you don't see my slide? Okay. Sorry. Did you see my slide or not? I just see a message.
Actually, your slides are not moving, doctor. Can you make it full screen and then you can.
You didn't see my slide from the beginning. Oh, sorry.
We see your slide, but not moving.
Okay. So I'm really sorry. And now is it okay for you?
Yeah, moving. Okay.
I'm sorry. Do you want that let me show you maybe. Yep. So it's okay for you now?
Yes.
Yes.
Great.
Oh, my side.
I'm sorry. So the strategy that I showed before is this one. So the interest of using particle as template and the interest of using the layer by layer technique. So as I said, I just want to show you the interest of using such strategy that I described in few words here for specific biological application. So as I said, we are collaborating with the team of Professor Gascon. So she's interested in finding a solution to treat pancreatic cancer. And yes, and the big problem of this pancreatic cancer, of this disease, is linked to the fact that it's diagnostic at an advanced stage, and currently there is a lack of effective treatment. So it's very important to find a new innovative treatment, more precisely, to find treatment which allow the targeting of this cancer cell. Interestingly, the team of my colleague have evidenced the possibility to use a pseudopeptide. So you can see here the chemical structure of the pseudopeptide and the interest of this pseudopeptide or as cytotoxic effect. From a physical-chemical point of view, the main interest for us is linked to the fact that this pseudopeptide is highly soluble in water, stable in biological media, and as you can see here, there are some positive charge. So it's a polycation. So we can use the strategy that I just showed you to prepare coated particle and also polyelectrolyte capsule. So we prepare particles from different size, from 20 nanometer to 100 nanometer. The great interest of this strategy is that we can control the amount of this pseudopeptide, and we can add this pseudopeptide as last layer. So please remember that this pseudopeptide allows the targeting that the polyelectrolyte capsule presents better biological activity than coated particle. I will talk again the interest of why we have such property of such impact of the particle property after. So then we developed in collaboration with the Dr. Manabe. So we developed more sophisticated template, which consists in using calcium carbonate particle and the cyclodextrin chemistry. So the calcium carbonate particles are very interesting because they are biocompatible, biodegradable, and we can easily prepare them from micro to nanometer range. The interest of cyclodextrin is linked to the fact that, as you may know, such saccharide allow the loading of hydrophobic compound. And by combining these two strategies, we prepare particles enabling the loading of two cytotoxic, one being hydrophobic and the unique one, so this pseudopeptide. I'm sorry, I cannot show you the chemical structure of the hydrophobic because we are trying to patent it. But the interest of this strategy is that these two cytotoxic has a synergetic effect, I mean the cells. Of course, we have then modified the surface of the particle by layer technique with a polycation and polyion that I cannot show you, sorry. Again, we are trying to patent the chemical structure of the different polyelectrolytes. And you can see here we use disulfide, so we develop redox responsive system, and the interest of using disulfide is linked to the fact that disulfide are extremely stable in the extracellular media, but are quickly reduced into thiol due to the presence of reductant, which called glutathione, in the intracellular medium. And interestingly, the amount of glutathione is higher in cancer cell than in the healthy cell. Okay, so now let me show you the interest of using the strategy to try to propose a solution to treat a specific kidney disease. So for this, we collaborate with the team of Professor Sali. More precisely, I collaborate. They want to load a specific therapeutic DNA. Why? Because my colleague would like to find a solution to treat the idiopathic nephrotic syndrome, so the kidney disease. So I just say a few words. Please remind that I am not a biologist, just a physical chemist, but it's important to understand the problem to try. So briefly, this kidney disease is related to the kidney membrane degradation. So as you can see here, there is a very simple representation of the kidney membrane of a healthy and a diseased one. And my colleague have evidence that this kidney membrane degradation is due to the overexpression of a protein. And they designed a specific therapeutic DNA which allow the inhibition of the protein expression. Unfortunately, as you may know, we cannot inject the DNA as it is because it can degrade by the nuclease and also does not incorporate into kidney cells. Moreover, we have to prepare particles with specific size, which is related to the porosity of the kidney barrier. Another thing, we have to prepare positively charged particle, and this is related to the DNA release mechanism inside the cell, because as soon as the particle is entered inside the cell, it can interact with the membrane of the endosome, which is negatively charged because the endosome membrane is composed by phospholipid, and this will lead to the endosome rupture. So we have to find first the good physical-chemical condition for the layer-by-layer assembly of the DNA and a polycation. As polycation, we have selected chitosan because chitosan is behave as polycation in some kidney disease. So for this, we use the quartz microbalance technique. So as you can see here, the interest of this technique is that according to the physical-chemical condition used, we can see in a real time the variation of the thickness and also the viscoelasticity property of the multilayer film. And when we find the good condition for the layer-by-layer assembly, we have simply to transpose this condition to modify a template. So here we select gold nanoparticle. The great interest of the gold nanoparticle is that we can easily prepare them or we can buy them. They are biocompatible, so we can modify the surface and check by zeta potential measurement that we have indeed the successful layer-by-layer assembly. Then we have studied the colloidal stability of the particle, and we see that they present good colloidal stability according to the time and the temperature. After that, we remove the template. So I say in the beginning that we can remove the template, but I didn't explain how. So we can, for instance, use this salt to etch the gold nanoparticle and to form this complex. So this complex will be then easily removed by dialysis process. And we have checked that in the capsule, we have not this complex. And as you can see here by TEM microscopy, we can show that we have indeed bare nanoparticle, modified particle, and capsule. So we have then adapt our strategy to prepare particles with different size, different number of layers, and all the particles have been analyzed by our colleague. So let me just show you one result that we obtained. We have shown, for instance, that our particles are able to inhibit the protein expression which induce the kidney disease, and we still see that the polychloride capsule presents better results than rigid particle. We of course we of course show that there is no toxicity of our particle, and we were very happy because this is the first DNA-based particle able to reach podocyte. So I mean, the first particle able to reach kidney cell, and we have patented this study. Okay, so now let me show you another example or another interest of using the particle that we are developing. For instance, we can use them as antibacterial system. So as you may know, there is a big problem due to the antibiotic resistance of some bacteria. There are very interesting systems which are reported in the literature and have been evidenced to allow or to try to find a good solution to antimicrobial resistance. The main mechanism is related to the fact that positively charged particles can induce bacterial death. So we try to propose an alternative to the existing system. And for instance, we show that we can use our system to load antibiotic. So for instance, if we use antibiotic which can present electrostatic interaction, so for instance, penicillin, we show that we can control the amount of antibiotic according to the number of the layer or according to the particle size. And then antibiotic-loaded capsule, again, pathogenic bacteria, and we have evidence that there is both an inhibition of bacterial growth and bactericidal effect. Interestingly, we have evidence that if we use particle without any antibiotic, we still observe an inhibition of the bacterial growth compared to particles which are coated. So again, it seems that we see an impact of the flexibility of the particle, and the exact mechanism how this flexible particle impacts bacterial growth is in progress. So now I would like to show you the interest of using the Boronic chemistry to study bacteria. I mean, the interest of developing glucose responsive system in this context. The first interest is that it's proven that the hyperglycemia induces bactericidal growth of the plaque. And the second point, it is shown that gram-negative bacteria present on the surface lipopolysaccharide. So we can use boronic acid to try to target such bacteria. Indeed, in few words, boronic are able to interact with diol or glucose to form this stable complex. And all this control according to the physicochemical condition study. So if we take a look at the literature, so if we see which are the already reported system, you can see that there are very interesting and very nice work reported by different colleagues. And in both cases, they have evidenced the total stabilization of the multilayer film up to a critical concentration of glucose. If we see the system, in most of the case, as I say, there is a total stabilization, and the one or two systems are composed by synthetic polymer. So the first thing that we have done in our team is to prepare multi-layer film entirely based on biopolymer. And we still observe a total stabilization of the multi-layer film up to critical concentration. I mean, if we prepare multi-layer film with this biopolymer and we load this film by drug, for instance, by insulin, up to a critical concentration, there is an immediate or quick release of insulin. Then we try to propose another approach based on swelling effect or a change in permeability. So for this, we prepare multilayer film based on chitosan and the boronic acid chemistry. This is based on the fact that if we take a look at the literature, if we prepare multilayer film on chitosan, and if we cross-link the multilayer film, it is evidence that there is a change of permeability according to the pH. And we have combined this idea with the boronic acid chemistry. I mean, we prepare alginate, we modify it with boronic. And interestingly, in this case, what we see is equal concentration of glucose, there is a swelling effect. And we can control this effect according to the average molar mass of chitosan, the cross-linking time, and the number of layers. So in this, there is a progressive release of the drug according to the time. So to conclude, I show you the interest of the strategies that we are developing based on templates. So we use particles that we can control the size, as I say. We modify the surface to prepare stimuli responsive system to allow a controlled release of the drug. The most important thing is to control the physicochemical condition of the particle to still keep the color. And I show you in some direct application the interest of the system that we are developing. All the results that I show you, we have published them or we have patented it. So if you want to have more information, please have a look on this paper, or please do not hesitate to ask questions. And of course, the funding for their contribution to this work. Of course, to thank you for your attention. I hope you have enjoyed this talk. Please do not hesitate to send me mail if you have questions or if you want to collaborate. So thank you very much for your attention.
Thank you so much, Dr. Sabrina, for joining us today. And we were glad to have you as a keynote speaker. And also, once again, I would like to thank you for understanding and our accommodating according to our schedule. And yes, if the participants have any question, they can go on and ask. And if not, they can text you on your email ID as well. In case there are no questions, I would like to proceed with our next presentation. Thank you once again, Dr. Sabrina.
Thank you very much. Have a nice day.
Okay, now I would like to call upon our next speaker. As we are now starting the poster session, e-poster session, I would like to call upon our first presenter, and that is Miss Maria from University of Medicine and Pharmacy of Craiova, Romania, and she'll be presenting on the topic PLGA-based biomaterials as drug delivery devices. Hello, Dr. Maria.
Thank you very much.
Yeah, you're most
Hello. Can you hear me?
Yeah, we can hear you.
Yes. Let me share my screen with you. Can you see the poster? It's okay?
No, actually, your screen is not visible to us.
Let me try again.
Okay.
Now?
No, actually, it's not visible to us still.
Okay. I will try again.
Now it's started sharing, but it's still showing blank. We should wait for some time.
Yes. Let me.
Yeah, we can hear you.
Okay.
Yeah.
Thank you for your fine introduction. My name is Maria Ciocâlteu. I am a postdoctoral researcher at University of Medicine and Pharmacy of Craiova, Romania. I would like to present a few words about some PLGA-based materials we synthesize in order to use them as drug delivery devices. As you may know, drug delivery systems are systems that introduce therapeutic agents into the body by controlling the rate, the time, and the place of release. PLGA, it's a synthetic copolymer with quite unique properties. First of all, it is a copolymer of lactic acid and glycolic acid. It is biocompatible and biodegradable. It suffers a hydrolysis process and decomposes in time into copolymers that are easily metabolized by human body, as I show in this picture. The ratio between lactic acid and glycolic acid influence the release of active principle and the degradation time. This is why we choose to work with PLGA with the ratio lactic acid to glycolic acid 65 to 35, so that we can have approximately 30 days release of the therapeutic agent. Also, it is accepted by Food and Drug Administration. The first medicine introduced in pharmaceutical market was used since 2002. It is called Lupron, and it has leuprolide acetate encapsulated in PLGA, and it is used to treat prostate cancer. After that, in 2006, naltrexone was used as opioid antagonist, and between 2002 to 22 and 23, the last year, 40 other new drugs, PLGA-based drugs, are in clinical trials. In our studies, we combine PLGA with antibiotics such as gentamicin, which it has several side effects, ototoxicity, nephrotoxicity, or antineoplasics such as doxorubicin, in order to obtain local release systems with lower toxicity. As a synthesis, we choose to use double emulsion, water, oil, water. In this method, first of all, you prepare a primary emulsion formed with aqueous phase of 5 milliliters, in which we dissolved the active principle, mixed by the oil phase, 100 milligrams of PLGA in 5 milliliters dichloromethane. The two phases were mixed in a vortex in order to obtain that primary emulsion. After that, we added drop by drop to a secondary aqueous phase with a huge volume, 95 milliliters, this primary emulsion, and stir for 3 hours in order to evaporate the dichloromethane, that it's a toxic solvent. The final suspension was centrifuged and subjected to a lyophilization process. After that, for those that contain doxorubicin, we try and succeed to surface functionalizate the PLGA with alendronate and zolendronate in order to enhance biocompatibility. We physicochemical characterized our nanoparticles by dynamic light scattering. We determined the encapsulation efficiency by HPLC. We studied the morphological aspects by scanning electron microscopy. And for those that contain gentamicin, we also studied the antibacterial activity. You have here a scheme
of our nanoparticles. Inside is the active principle that is hydrosoluble. This is a simple particle, and this one is functionalized with biphosphonates. By morphological characterization, we observe that no matter what therapeutic agent we have inside PLGA, we have a typical morphology, spherical one, with pores that help the drug release. We did also a numerical distribution of the particles using dynamic light scattering. In these images, you have PLGA functionalized with alendronate. And for those, we observed that the average of the particles is 87 nanometers. But for all the other compounds, composites we synthesized, we observed dimensions between 0 and maximum 315 nanometers. It is also important to mention that the zeta potential has negative values. For those one, it is with the value minus 42.09. This is good because it means that our composites do not form aggregates, they are stable. For those with gentamicin, the encapsulation percent was 20%. We determined that using HPLS with the mobile phase, an isocratic mixture of acetonitrile and sodium citrate solution. You have here the calibration curve. Gentamicin is not a single compound. There are 4 compounds inside it. So we decided to measure the encapsulation efficiency by peak number 4, this one. And this is a typical chromatogram for our composite. It is good that even at this percent that may be small, we have a significant antibacterial activity. And as you can see from the diameters on the inhibition zones, the standard gentamicin and PLGA gentamicin samples have similar activities, even if we put in one of our compounds hydroxyapatite to improve the mechanical strength of our composites. In conclusion, the PLGA-based biocomposite nanomaterials are efficient as drug delivery devices in multiple therapeutic agents. The advantages of such a biomaterial target the health system, being easy to obtain through a cost-effective method, and PLGA remains the smart biopolymer targeted drug delivery research. Thank you very much.
Hello, Dr. Maria. We can't see the screen now. Have you completed your presentation?
Yes.
Okay, I'm so sorry. And thank you so much for your presentation. And if anyone have any questions, they can go on and ask. Are there any questions for Dr. Maria? In case there are no questions, I would like to thank you once again, Maria. I would like to move forward with our next poster presentation. Now I would like to call upon Mr. Siddhant Saxena from Polytechnic University of Catalonia, Spain, and he'll be presenting his poster on study of self-assembly behavior of beta-cyclodextrin and its functionalized derivatives. Hello, Siddhant, are you there? Can you listen me?
Yes, yes. Hello, can you
Yeah.
hear me?
Yeah, we can hear you.
Good afternoon. Yeah, I can hear you. Yeah.
Okay.
I'm going to share my screen. Is it visible? Is my poster visible now?
No, actually, try opening your post in your computer. Actually, we can see the screen of the meeting.
Okay, wait. Now is it visible?
Yes, yes, you can start.
Okay, so good afternoon, everyone. My name is Siddhant Saxena, and I'm a doctoral student in Polytechnic University of Catalonia, Spain. And today I'm going to discuss about the self-assembly behavior of beta-cyclodextrin and its functionalized derivatives that we have used. So the main aim of this study was to understand the aggregation in beta-cyclodextrin and in sulfobutyl ether and hydroxypropyl beta-cyclodextrin. So what we have tried and why we are doing this is because if you will see that beta-cyclodextrin has been widely used in pharmaceuticals for a long time now, but no one has actually focused a lot about the self-aggregation behavior of beta-cyclodextrin. So we thought of doing this study. So firstly, we should know that beta-cyclodextrin is a cyclic heptasaccharide. So it contains of 7 glucose units, which are joined in a ring shape, as you can see here in the structure. And it is among the 3 naturally obtained cyclodextrins, along with alpha, which contains 6 glucose unit, and gamma, which contains 8. So among the alpha, beta, and gamma, beta-cyclodextrin shows a very poor water solubility. The reason being the formation of the intramolecular hydrogen bonding between the carbon 2 and the carbon 3 of the glucose unit. So there is this intramolecular hydrogen bonding, a belt of intramolecular hydrogen bonding, which restricts the beta-cyclodextrin from being solubilized in water a lot as comparison to alpha and gamma. And this also leads to the crystalline structure of the beta-cyclodextrin in solid state, as well as the low solubility leads to the aggregation of the beta-cyclodextrin in water. And that is what we have tried to study. So here, if you can see the structure, the truncated cone structure of the beta-cyclodextrin, there are total 21 hydroxyl groups. 7 of them are primary, 14 of them are secondary in nature. The secondary hydroxyl group, as I told, form the hydrogen bonding. The primary hydroxyl group are outwards, and they are responsible for somewhat solubility of the beta-cyclodextrin that we see. We tried to see the aggregation behavior using DLS, optical microscopy. So here you can see that we saw an increase in the aggregate size with respect to concentration as well as with respect to time. That was very evident in the case of beta-cyclodextrin because some of the researches has already shown that beta-cyclodextrin tends to aggregate. But when we saw the results in the case of sulfobutyl ether and hydroxypropyl beta-cyclodextrin, we were surprised because they also tend to show aggregating behavior. In sulfobutyl ether, we found out that the SO3 minus moiety, which is there, the electronegative moiety, so the repulsion between the negative moiety is not enough that it can stop the aggregation of beta-cyclodextrin. This we confirm through the values of the zeta potential as well as through the molar conductivity. In the case of hydroxypropyl again, we saw a increase in the solubility as compared to native beta-cyclodextrin. because of the disruption of that secondary hydrogen bonding that was present. But again, here we also saw some sort of aggregation process with time and well as with concentration. Then we tried to compare the aggregation in the beta-cyclodextrin, sulfur-butyl ether beta-cyclodextrin, and hydroxypropyl beta-cyclodextrin. And we saw that with time, beta-cyclodextrin was undoubtedly aggregating more as compared to its functionalized derivatives. And we calculated the CAC, you can see here. The CAC in beta-cyclodextrin was 9.8 millimolar, which is around 1.11% weight by volume, as comparison to the sulfur-butyl ether and hydroxypropyl beta-cyclodextrin. We also perform the optical microscopy just to see that how the aggregation is growing, like how this self-assembly process is following an order. So here you can see that at lower concentrations, for example, in sulfur-butyl ether, we are seeing a small fiber type of structure, right? But when you see this at higher concentration, around 2.5% weight by volume, you saw this, that the fibers have thickened and there are some sort of nodules which are being formed. So one thing we can confirm from both of the cases, as well as in the beta-cyclodextrin, you can see here, that the growth is in a worm-like fashion. It's not something kind of a big like chunk of aggregate being formed. No, the growth is in like a fibril form, although the thickness of that fiber increases with the increases in concentration. So yeah, all in all, we can come to the conclusion. So what we saw is that native beta-cyclodextrin has more tendency to grow aggregates as comparison to its functionalized derivatives. But we also found out at the same time that functionalized derivatives also aggregate, although less than the native one, but they aggregate. So they do have a tendency to aggregate. We saw that the aggregation happens in a worm-like fashion. The growth of the aggregation is in a worm-like fashion. Now, why we did this research and why this research is important? You might have seen the cases where beta-cyclodextrin is being used to form inclusion complexes or metal organic frameworks. So we want to check that does the self-assembly behavior have any effect on the, for example, stability or on the final structure of these type of complexes that we are making or not. And you can also see that beta-cyclodextrin has been highly used in pharmaceuticals for drug solubilization. So we again wanted to check that is the self-assembly behavior of beta-cyclodextrin going to affect the drug solubility? Yes or no? So these were the basic motives to, and that is why we understood that how the self-assembly behavior of beta-cyclodextrin works. This research is the basic research and it can be used as a comparison to some of the forward researches that are being done by the people. People can try to understand the self-assembly behavior of beta-cyclodextrin and then try to compare it with the with their research, that how the self-assembly behavior is going to affect their final structure that they are going to make. Thank you so much. That was it from my side. Hello, am I audible?
Yeah, you are audible.
Yeah. I've completed.
Okay, thank you, Mr. Sudan, for your presentation. Do anyone in the audience have any questions? Okay. Okay. Thank you once again. And now we would like to proceed to our next presentation. Okay. I would now like to call upon Dr. Andrea Vita from Bioboron Research Institute, Romania, and she will be presenting a topic on ZNB PLGA nanocomposite material for biomedical applications. Hello, Dr. Andrea, are you there?
Hello, can you hear me?
Yes, we can hear you.
Perfect. Thank you for the introduction. So yeah. I will try to, if you can give me one moment to.
Yeah, sure, take it.
My screen. Is it visible?
Yes, it is visible.
Yes, perfect. So good afternoon, everybody. My name is Andrei Bita from the Bioboron Research Institute in Craiova. My presentation is about another PLGA nanocomposite. This time it is a different type of compound that we integrate in these nanocomposite materials. We actually work with the Faculty of Pharmacy. My colleague who previously mentioned these types of nanocomposite materials with the PLGA, we actually work with them, and they help us to put these types of compounds that we synthesize inside the PLGA nanocomposite materials. So regarding the introduction, I will not enter into much detail. My colleague previously mentioned about the general part of the PLGA copolymer. So in general, we used basically the same ratio of copolymers, 65-35 lactic to glycolic acid. So this doesn't change the, let's say, degradation or delivery period of the compound, which is pretty much the same for the antibiotics she mentioned earlier. What we actually try to insert in this type of PLGA nanocomposite materials is, let's say, a new synthesized sugar borate complex, which was functionalized with zinc. So we have some experience in this, let's say, sector with the sugar borate esters. We try to synthesize them for some time now, and we try to functionalize them with different types of not only metal cations but any other types of cations. So why zinc and why boron? So recently boron has been found to have interesting impact on the health of the microbiome. So this is an interesting, this could be an interesting, let's say, drug for future uses in different types of pathologies that have consequence from different types of dysbiosis. And also zinc, which is an essential trace element, it also can have an impact on the microbiome, but it is also involved in different other types of mechanisms in the body, like let's say glutaminergic transmission as a signaling molecule. In the antioxidant response, it can be a cofactor in many enzyme and transcription factors. So an abnormal zinc homeostasis can cause a variety of health issues, including growth retardation, immunodeficiency, hypogonadism, neuronal and sensory dysfunctions. So this is why we try to focus on, let's say, this type of compound. The synthesis of the PLGA composite is relatively similar to what my colleague previously mentioned. So we have an aqueous phase in which we dissolved 50 milligrams of this zinc boron sugar complex in 2 milliliters of aqueous solution with 4% polyvinyl alcohol. So the oil phase was 50 to 150 milligrams of 65 to 35 PLGA, which was dissolved in dichloromethane. The two phases were brought together into contact and mixed at high RPM 45,000 in order to obtain this primary emulsion. At the end, the primary emulsion was dripped in a large volume of aqueous solution of 95 milliliters with 1 to 4% polyvinyl alcohol as an emulsifier. This one was stirred at around 1,000 RPM in order to get rid or evaporate as much of the dichloromethane as possible. So after the synthesis, we actually freeze-dried, lyophilized the nanoparticles, and they were physicochemically characterized by a series of assays. So maybe the first one was the scanning electron microscopy, at different magnitudes, it revealed a porous material, yes, that was formed in various spherical shapes, which was the zinc boron PLG biocomposite. It was very well organized, and the three-dimensional aspect is actually highlighted here in the SEM figure. The hydrodynamic diameter of the particles was assayed as well. We obtained two types of granulometric intervals, one between 190 and 291 nanometers, and the other one between 1 and 1.6 micrometers. But the majority of them were around 225 nanometers. Also, the zeta potential was negative with a value of -5, not as low as the particles that were obtained previously by my colleague, but nonetheless a negative value for our nanoparticles. To conclude, this method could be extremely well used to obtain this type of nanocomposite material complex, and to administer this type of compound, zinc boron complex, to treat further pathologies that can have their origin in microbiome dysbiosis or other type of zinc deficiency. Limitation of our study would be, but we are in the works for this. We didn't have or we didn't manage yet to identify the actual load capacity of the nanoparticles with the zinc boron complex. So this is in the works, and probably most probably the results will be published in the near future. Thank you very much for listening. So can you hear me?
Yes, doctor, we can hear you.
Thank you.
Yeah, thank you for presenting.
Thank you.
Yeah, if any of the participants have any questions, they can feel free to ask. If not, we can move to our next presentation. Okay, thank you once again, doctor. Now I'd like to move to our next presentation. Now I'd like to call upon Dr. Nehla from British.
Yes, hello.
Yeah, hi, most welcome. And she yes, you're audible, and your slide is also visible to us.
Okay, the poster is clear.
Pardon, please.
Everything's clear. Shall I start?
Yeah, it's clear. You can start.
Okay, thank you. Hello, everyone. My name is Nahla Al-Hasisi. I'm a lecturer assistant faculty of pharmacy, the British University in Egypt, Cairo, Egypt. And my topic today is about 3D nanofiber scaffolds for posting the repurposed dermatological effect of spironolactone. Let's have a brief introduction about spironolactone. So what's a spironolactone? Spironolactone is an aldosterone antagonist that has anti-androgenic properties. Its FDA-approved use is commonly used for treatment of arterial hypertension, heart failure, primary hyperaldosteronism, and edema associated with post-nephrotic syndrome and liver cirrhosis. But besides the commonly used purposes for spironolactone, it also has a great efficacy towards repurposed dermatological diseases such as rosacea and acne. So let's have a brief about what is repurposing or repositioning. Actually, repurposing, repositioning, reprofiling, or even retasking is a strategy in which new uses is already found for an already approved drug. So what could be the advantage of repurposing? Actually, it's a shorter time strategy for drug development. And we all noticed that during COVID-19 pandemic, it's a less expensive strategy and work on overcoming the inflation cost of drug development and ultimately reducing the actual cost of therapy. So what could be the relationship between nanotechnology and repurposing? Actually, nano reformulation of repurposed drug is a great approach through which we can maximize the repurposed use of drug and together with minimizing any unwanted side effects. So the aim of work is to overcome spironolactone dermatological uses challenge via its reformulation into topical nanofiber scaffolds. So let's have a brief about what's rosacea disease and what's acne. Actually, rosacea is a chronic multifactorial skin disease that primarily affects the face and characterized by facial flushing. It also has an ocular involvement such as conjunctivitis or pellipheritis. And acne, all of us know acne. Acne is a skin disease of pilosebaceous unit, and it's commonly found in face but also present in chest, upper back, and neck called body acne. So our dosage form selected in this study was nanofiber. So what's nanofiber? Nanofiber is one of the most promising and universal drug delivery systems that can be tailored for a wide range of drug release kinetics. It consists of solid fiber with diameter size starting from few nanometer to 1000 nanometer with multiple unique functionalities and characteristics. Nanofiber advantage as it is one of the most promising therapeutic approach to deliver drugs to skin in more precise and accurate manner. It also works in improving skin quality and targeting many skin-related diseases. It also has many advantages such as high drug loading capacity, high surface area, ease of functionalization, cost effectiveness, and adequate mechanical strength. It also can achieve both immediate or modified release and can be administered through different routes of administration. To prepare nanofibers, the method of preparation of its choice is electrospinning. Electrospinning is a scalable method which produces 3D nanofiber with unique characteristics that facilitate its use in industrial stage. In this study, we created two scaffolds using two different polymers, which is polyvinyl pyridine polymer and polyepsilon caprolactone. Polyvinyl pyridine polymer is a chemically stable polymer with an inertness, lack of irritation to biological system, low toxicity, and biocompatibility. The optimized former from PVB nanofiber were further loaded with spironolactone, and it was subjected into in vivo model to be tested for rosacea effect. And the BCL, which is polycaprolactone, have also advantages being biocompatible, biodegradable, thermally stable, chemically stable, and good mechanical strength. After optimizing BCL nanofiber, it was also loaded with spironolactone, and the fabricated scaffold were characterized and further subjected into in vivo model, which is to discuss its anti-acne improvement. So what could be the potential advantage of loading spironolactone into nanofiber scaffolds? Actually, we work in decreasing the line of treatment using oral antibiotic for those diseases as to minimize antibiotic resistant dilemma. Also, to minimize the severe systemic side effect, as I mentioned before, of oral spironolactone, and some patients decide to use some cosmetics to conceal the symptoms of either rosacea or acne, but those phase worsen in the case by time. Also, the topical non-antibiotic treatment may cause friction during rubbing that exaggerates the sensitivity skin during application for rosacea patients. So, in our study, we optimized some factors such as low, medium, and high concentration of PVB polymer, and the optimized one were further loaded with spironolactone. Regarding BCL, we tested two items, which is the molecular weight of BCL. We studied low and high molecular weight of BCL, and also studied different solvent system, and the optimized one were also further loaded with spironolactone. Our characterization was varied starting from making and scanning electron microscope to see the morphology, FTIR, XRD, also AFM, water contact angle, and swelling test. The in vitro release test was done using total immersion method, in which the specific weight of the nanofiber scaffolds was immersed in phosphate buffer saline, pH 7.4, and sample were withdrawn and analyzed spectrophotometrically. The ex vivo test was also conducted, and moving to the in vivo test. We conducted two in vivo tests, which is croton oil rosacea model for testing the anti-rosacea efficacy of PVB scaffolds after loading spironolactone on it. And also we conducted testosterone-induced acne model. Let's now have a brief about our results. PVB showed that neither low molecular weight or high molecular weight polymeric solution can neither low concentration or high concentration polymeric solution could perform the optimized nanofiber, but the selected one was the medium PVB concentration which maintained the smoothness of the fiber after loading spironolactone on it. For BCL nanofiber, the low molecular weight BCL couldn't evenly produce any kind of nanofiber either with changing the solvent system. While after using the high molecular weight BCL, nanofiber started to appear, but the quality changed from being optimized using mixed solvent system, chloroform and methanol, not chloroform. And as we can see here, how the fiber is increased. FTIR and XRD showed that, first let me start by FTIR. It showed the no kind of interaction between either BVB or BCL with the drug and indicates the compatibility between the selected polymer and the drug. XRD showed the change from crystalline into amorphous change after electrospinning, which indicate the successful incorporation into nanoparticles. We found that there is a difference in the release pattern between SPBVB nanofiber and SPBCL nanofiber. For SPBVB nanofiber, it started by very fast release followed by a bit controlled area up to 24 hours. In contrast with BCL nanofiber after loading with spironolactone, it showed that very slow first release and more controlled release up to 180 hour of release. The in vivo model also showed no inflammation for ears of animals after treatment with SP-loaded BVB nanofiber. And regarding the acne, it showed also the reduced number of sebaceous globule and sebaceous number size in pilosebaceous unit for SPBCL nanofiber. Moving to the conclusion, we could conclude multiple items such as that nanofiber have a great potential to enhance drugs efficacy via a different route of administration, and the topical delivery of spironolactone through nanofiber is a promising, successful technique that can be a great carrier for both anti-rosacea and acne and anti-acne repurposed uses. We can also say that synchronization between nanotechnology and repurposing strategy is a great approach with multiple win situations. Thank you for your listening and ready for any questions.
Hello, Dr. Naila. Thank you for presenting.
Hello?
Yeah. Hello, doctor.
Yes, hello, sir.
Yeah.
Madam.
Thank you for presenting. Now, if any of the participants have any questions, they can ask. Okay, and if there are no questions, I would like to.
Okay, thank you all. Thank you so much.
Now I would like to call upon our next poster presenter, Dr. Sara from University of Florence, Italy, and she will be presenting her poster on development of cross-linked protein-based materials from vegetable sources. Hello, Dr. Sara, are you there? Can you listen? Can you hear me?
Yes.
Yeah, most welcome.
Can you hear me?
Yes, we can hear you.
Okay, thank you. I'm going to share my screen. Can you see it?
Yeah,
okay. I'm sorry, I have some problem of connection from this morning. I'm very sorry, but I hope that you can see me correctly.
Yeah.
Okay, thank you. Thank you for your nice presentation. I'm Sara Guiglia, and I'm a PhD student from the University of Florence. And I'm going to present you part of my PhD project. This PhD project is developed in collaboration with a company. The name is Pimpet, and is a company from Pisa. My project is the first step toward the development of a biopolymer in order to obtain a fully sustainable material. for multiple applications. Indeed, in recent years, there is increasing interest to the development, to use natural polymers to obtain material that can replace totally, if impossible, but in part, the petrol-based material. Between these natural biopolymers, protein has reached an interest because of their availability from animal or vegetable waste. In particular, in my project, we decide to use protein from rapeseed bioproduct as our main ingredient of our material. The rapeseed, and particularly, we decide to use the rapeseed meal. That is a bioproduct obtained after the oil extraction from the rapeseed seed, and it is obtained after mechanical extraction and ethane extraction. This meal is rich in protein. Indeed, the protein content is about 40%, and it has good properties and promising properties to obtain a material. Indeed, is product in high amount for here, particularly in Canada and USA, but also in Europe. And it has film properties and high oxygen and carbon anhydride resistance. Particularly, we start by the extraction of the peptide contained in this meal, and I did two different extraction in two different pH, particularly at alkaline pH of 8 and in acidic pH of 3. And I performed this extraction at 60 degrees and for 4 hours. From the XLC chromatogram, we can see that I could extract different peptide that has FARP hydrophilic peptide. And also we can see that from the alkaline extraction, we have more peptide than in the acidic extraction. Also, I did the proximate analysis of this extraction, and we see that the protein content is the 21% 0.75 for the alkaline extraction and 18.91% in the acidic extract. And also we can demonstrate that this extraction is reproducible, easy to scale up. Indeed, this extraction was did at scale of 50 grams. That is a high laboratory scale, and is also eco-friendly because we did this extraction without the addition of other additive, just using sodium hydroxide in case of the alkaline extraction, and chloride and HCl in case of the acidic extraction. Then we studied the property of the rapeseed meal to obtain a material, and we mix it in a blender blade the canola meal with glycerol, water, and another protein-based compatibilizer. And we study the capacity of the protein chain to cross-link each other in order to obtain a material that is that could have better mechanical properties. And we did the formation of cross-linker with the eboside. Unfortunately, I cannot say more detail because my project is in collaboration with an agency. But what we want to do is to cross-link the protein chain the cross-link used the positive cross-link that react with the amino functionality of the lysine. This is in 2.26% in the rapeseed meal protein. And we process this mixture using a flat blend press at 150 degrees for 15 minutes at 2050 bar of pressure. And once obtained our material, we saw from this diagram that had the stress strain diagram that the material has the promising mechanical properties. Indeed, it is flexible and also resistant to the deformation. And once obtained our prototype, we try to improve the mechanical properties using the water mixture of reductive agent, the natural agent, or both together. Indeed, the reduct agent broke the sulfide bridge bond in the protein. It can disrupt the quaternary structure of the property, help the cross-linker to go inside the protein and react with the lysine. And what we see when we use the reactive agent, the elastic module increases, and it can be explained by the formation of the cross-linking, because the cross-linking information help the material to be more brittle and more resistant. And the same behavior, we have the same behavior with the natural agent. The function of the natural agent is to disrupt again the quaternary structure of the protein and help the cross-linker to react with lysine. And in this case, again, we saw an increase of the elastic module, but also an increase of the deformation before the break of the material. So we decided to use the sodium sulfate and the guanidine together to show if the mechanical property could be better again. Could be better. And what we see when we decide to use the sodium sulfate at 10% with guanidinium in 7% and in 14% in the total mass of the material. In this way, we introduce more water that as all of you know, the water have work as plastificant in biopolymer. And also we decided to use the sodium sulfide at 20% with ganidimon, again at 7 and 14% in the total pass of the material. And then we saw the better mechanical properties. So we studied the water absorption of this material, and unfortunately, as you can see from the diagram, this material absorbs high content of water, and also after when we calculate the soluble material loss, we saw that we have a loss of the 30% of the mass that could be the glycerol that migrate in the material, and it is a common problem of the glycerol. Therefore, we'll try to use the peptide extract together or in place of the glycerol to see if this extract can work as plastificant thanks to their compatibility to the protein chain, and form more stronger hydrogen bond and van der Waals bond, and in order to decrease the loss of the plastificant. However, from the stress strain test, we saw that this material works as plastificant. Indeed, when we use the 100% of extract, of 50% of the extract, and 50% of glycerol, we have a high elastic modulus than the static material, in which we have the 20% of sodium sulfite and the guanidine in 14%. What we can see in this project, that this material is very promising, even if it needs more studies in order to attain a material that could be commerciable. And thanks all for your attention, and thanks to the organized committee and for this opportunity to participate at this nice conference. Hello, can you see me? Hello?
Hello, Dr. Sara. Thank you for presenting with us today, and we were glad to have you. Also, if any of the participants having any question, they can ask. As we have already completed all our presentations for today, and the remaining speakers that were supposed to join us today, we are not able to join due to some personal reasons, or maybe because of their internet connectivity. But I hope we all have enjoyed today's virtual session. Thank you once again, all of the presenters and all of the participants as well. I hope you had a good time and a great session, and I expect all of you are getting all of you got a good experience from our virtual session. Regarding the certificates, which we will be sending it by tomorrow. And now, if anyone is open for live discussion, they can feel free to go.
Thank you for your effort. Thank you all. Thank you. My pleasure meeting you all.
Thank you, Dr. Mela.
Thank you.
Thank you, Dr. Sarah.
You all can find my contact in the abstract, and you'd like to ask the organizer, where can I find the abstract? Is it published in the website?
Yeah, when I will be sending you the certificates, I will also make sure to attach you the book of abstract or the proceedings along with that, and there you can find your abstracts.
Okay. For all my respected colleagues, you can find my email in the abstract, and you can contact me anytime. Anytime you can visit Egypt, and you're more than welcome. Thank you all.
Thank you all.
Thank you all and have a great day.